Om Shri Sai Ram. My humble pranams in the lotus feet of Bhagwan Shri Sathya Sai Baba. Honorable Chief Guest, Dr. Raj Narayan Tiwari, Director, National Institute for Research in Environmental Health. Respected Dr. Meena Pimplapure, Chairman, College Governing Body. Respected Dr. Rajan Dubey, Chief Global Strategy at SRS Life Sciences Healthcare Scientist Singapore. Respected Principal Dr. Asha Agarwal, Vice Principal Dr. Jyotsna Galgale, Honorable Members of the Advisory Committee, respected Dr. R.B. Sharma, respected Dr. Abha Bajpai, learned delegates from across the globe, academia, participants who have joined online, and dear students. Good morning to all of you. I, Dr. Megha Singh, Assistant Professor English, on behalf of the Sathya Sai College for Women Bhopal, take the privilege to welcome you all on this 2 days international conference on post-COVID trends of biological sciences towards research, therapeutic, innovation, and entrepreneurship, organized by the Department of Zoology and Biotechnology, the Sathya Sai College for Women Bhopal, and is sponsored by MP Council of Science and Technology Bhopal. National anthem involves emotions and its pride for the country. Honoring the anthem is equal to honoring the nation. It generates the feeling of dedication and devotion towards every action we do. To begin with, may I now request our esteemed guests and participants to please stand up for the national anthem to show respect for our nation, believing and being confident that every action of ours will contribute to the progress and well-being of the nation.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you, everyone. I hope we all are now charged and encouraged in whatever endeavors we put in. I now take the privilege to invite our esteemed guest, Dr. Raj Narayan Tiwari, to kindly grace the dais. I also request Principal Dr. Asha Agarwal, Vice Principal Dr. Jyotsna Galgale, Convener Dr. Rupa Guha Nandi, and Co-Convener Dr. Sunita Yadav to accompany our guest. We are extremely grateful to our esteemed guests for showing benevolence and gracing the occasion with their incredible presence. Moving further, may I now request our guests to offer flowers to Goddess of Wisdom and Knowledge, Ma Saraswati.
जय जय देवी चराचर सारे पूज्य जुग शोभित मुख ताहारे पुस्तक हस्ते भगवती भाव
Father, we pray for your blessings and with all humility, give us wisdom to make this world a better place. May all human beings live in harmony with the nature and other forms of life. Thank you, sir. Thank you, madam. It is said someone is sitting in a shade today because someone else planted a tree long time ago. All our wisdom is stored in trees. We respect the thought and following the tradition of our institution, welcome our guests with saplings and convey a message that we care. May I now request our principal, Dr. Asha Agrawal, to welcome our chief guest, Dr. Rajnarayan Tiwari, with a sapling. Thank you, madam. We give a warm and heartfelt welcome to Dr. Meena Pimplapure, Chairman, College Governing Body, and our keynote addressee, Dr. Rajan Dubey, who have joined us online. We thank both of you for your time and gracious presence. I now invite our Vice Principal, Dr. Jyotsna Galgale, to welcome our Principal, Dr. Asha Agrawal, with a sapling. Thank you, madam. Dr. Rupa Gohanandi, convener of this conference, is requested to welcome our Vice Principal, Dr. Jyotsna Galgale, with a sapling. Thank you, Dr. Nandi. May I now request Dr. Sunita Yadav, co-convener of this conference, to welcome the convener, Dr. Rupa Nandi, with a sapling. Thank you, Dr. Yadav. Dr. Renu Srivastava, Assistant Professor, Zoology and Organizing Secretary of this conference, is requested to welcome Dr. Sunita Yadav, co-convener of the conference. Thank you, Dr. Srivastava. Touching hearts and transforming into excellence, emphasizing on transforming the learners to excel mentally, physically, technically, emotionally, and spiritually with love, care, and affection is the objective of our college. And such an objective cannot be achieved without a competent leader and head. I now take the privilege to invite the head of our institution, our principal, Dr. Asha Agrawal, madam, for welcome address. Madam, please.
Om Shri Sai Ram. My pranam in the lotus feet of Shri Sathya Sai Babaji. And I welcome our Honorable Chief Guest, Dr. Rajnarayan Tiwari ji, Director, National Institute for Research in Environmental Health, ICMR Niri, Bhopal. I also welcome Dr. Ranjan Dubey, Chief Global Strategy at SRS Life Science Healthcare, Singapore. And here I welcome our respected chairman, Dr. Meena Pimplapure, and all the distinguished speakers, esteemed guests, my colleagues, teachers, and dear students. It's an immense pleasure to welcome you all to the International Conference on Post-COVID Trends of Biological Science towards Research, Therapeutics, Innovation, and Entrepreneurship. I wholeheartedly welcome all the delegates across the world. After the current pandemic scenario, we all have gathered here to discuss the COVID-19 and its various effects on our world. COVID-19 pandemic has led to a dramatic loss of human life worldwide and presents an unprecedented challenge to public health, food system, and the working lifestyle in the world. Besides social disruption, which is devastating, this conference throws a light on the biological trends towards, towards research, therapeutics, innovation, and entrepreneurship. The crisis has reshaped the science and technology. It has led to fundamental changes in the physiology of human beings, experimentation with new tools favoring the life they have started at a very fast rate. I welcome the most distinguished speakers in the program who will discuss and highlight the key points, trends, and reforms in the system for the audience. It will be great opportunity to hear from our learned keynote speakers, Dr. Jasolova from Singapore, Lazana, Siberia, sorry, and Dr. Sumit Sachdev, Dr. Prashant Sharma, Dr. Manoj Kumar Das, Dr. Yusuf Akhtar, Dr. D.N. Ganesh, Dr. Sugandha Singh, and others. So it will be a great opportunity to hear from our learned keynote speakers about the influence, priorities, development, and compassion to international standards in the same field. My best wishes to the convener of the conference, Dr. Rupa Guhanandi, and convener, and Dr. Sunita Yadav, co-convener and organizing secretary, Dr. Renu Srivastava. The organizing committee members and volunteers of this international conference who have worked 24 hours and arranged such an enlightening, meticulous program. So in the end, I would like to say that health is wealth. So first care for your health, then care for your wealth. Don't put unnecessary thing in the nature and pollute the environment. And don't play with viruses too. Means microbiologists, they are there to work, but don't play with virus. Be careful while doing research on viruses. So Sairam and all the best to all. Thank you.
Thank you so much, madam, for your blessings and best wishes. The COVID-19 pandemic has sent shockwaves through societies and economies around the world. The impacts of the disease and of measures to control it have raised questions about epidemic preparedness and more generally about development, past, present, and future. Moving beyond the plethora of fragmented opinions and commentaries on various issues sparked by COVID-19, the pandemic has unfolded new areas of research and innovations. Post-pandemic transformations are thought about and enacted upon. As COVID-19 pandemic has charted its unprecedented path around the world, it has carried with it the question, what will COVID-19's legacy be? From healthcare to education, to entertainment to manufacturing, technology innovators are stepping towards to help answer that question. Therefore, it can be said, crisis can be a catalyst or can speed up changes that are on the way. It can almost serve as an accelerant. Pandemic COVID-19 has also brought challenges and exciting opportunities in the field of education and research too. It has given researchers new insights and wonderful solutions to the problems that have never been imagined of. The framework of this conference is also woven around the post-COVID trends in research, innovation, therapeutic, and entrepreneurship in the context of biological sciences, with a notion that quality is not an accident. It is always the result of high intentions, sincere efforts, intelligent direction, and skillful execution with dedicated team. The concept of this conference is designed. May I now invite Dr. Rupa Guhanandi, convener of this conference, to highlight the objectives of this 2 days international conference and orient the participants with the outline and expected outcomes of the conference.
Thank you, Dr. Megha Singh. Om Shri Sai Ram. I offer my salutation towards the divine energy who exists in all beings in the form of intelligence, mercy, beauty. My salutation is towards my beloved parents, my family, and also respectful salutation towards our chief guest, my dear teachers, dignitaries, and colleagues. My greetings towards researchers and dear students. On behalf of Sri Sathya Sai College for Women, I, Dr. Rupa Guanandi, would like to start my wishing you and your families my personal best for your health and safety in this era after pandemic. I would also like to express my profound gratitude to all those doctors, nurses, and workers who were serving so selflessly on the front line during health crisis. As we are in the midst of a crisis like no other, the pandemic has sent shock waves. Describes as alarming and part because it has served new and unfamiliar features. A global medical emergency caused by a virus we do not fully understand actually. During pandemic, the way people express grief over the death of their near and dear ones has changed. People mourn without relatives, friends, who would have come to lighten the burden of grief. I would like to express my sincere condolence towards those souls who left us in this pandemic. The main aim of this conference is for the betterment of all the students and their future scope in biological sciences after pandemic. This conference will create consortium in the area of advanced research, innovation, therapeutic, and entrepreneurship in biological sciences during post-COVID. Dignitaries, scientists, researchers, professors from recognized institutions, organizations from different areas, and students are going to attend this conference. I welcome all the speakers who will be with us in this 2 days conference virtually to share their valuable knowledge. Now, allow me to give a brief update of today's conference, which will run for 2 days with 4 sessions, excluding inaugural and valedictory. The session will consist of therapeutic innovation, entrepreneur, and research focused on the current post-COVID scenario in the field of biological sciences and their fields as well as advancement required in this era after pandemic. We have arranged the paper and poster presentation sessions in both the days of this conference. It will be an opportunity for us to listen from our phenomenal judges to put forward their reforming thoughts in this vast fields. This meeting will definitely bring together interdisciplinary science, researchers, students, and faculties working several fields of healthcare, biotechnology, microbiology, IT, computational medicine, chemical engineering, commerce, and facilitated an exchange of ideas that could lead to the recognition of effective research and technology for a healthy tomorrow. We all understand the importance of science, technology, innovation, and entrepreneurship in our daily lives, and the way in which they are transforming the world. I believe that by participating in this international conference, we are all in the right place in the right time. Together, let us accelerate the exchange of ideas and sealing up of good practices. I welcome you all again in this 2 days international conference in hybrid mode. Sincere thanks to all of you. Now you people can please cheer. आप लोग बहुत ज़्यादा दुखी दिख रहे हैं मुझे. Please cheer. आफ्टर पेंडेमिक हम चेयर कर सकते हैं. हमने ये इंटरनेशनल कॉन्फ्रेंस इसलिए बुलवाया है ताकि हम फ्यूचर में इस तरह का कोई भी पेंडेमिक आपके सामने आए तो हम इससे कैसे लड़ सके. अगर हम एक भी बीज बो पाए तो हमारे लिए एक सक्सेस रहेगा ये कॉन्फ्रेंस. थैंक यू ऑल ऑफ यू.
Thank you, Dr. Nandi. Can we all give a huge round of applause for all of us and make this environment full of energy. Proceeding further in the program, we are privileged to have an eminent person, Dr. Rajnarayan Tiwari, as our esteemed chief guest. I feel honored to present his bio brief before inviting him on for his address. Dr. Rajnarayan Tiwari is Director, ICMR, National Institute for Research in Environmental Health, Bhopal, a community physician by education. He did his PhD in life sciences and has an experience of 29 years in health research and teaching. He has many national and international awards and fellowship to his credit, including Dr. M.K. Seshadri Award by Indian Council of Medical Research, New Delhi, Honorary Research Fellow in Industrial Health, elected Fellow of Indian Association of Preventive and Social Medicine, elected Fellow of Indian Public Health Association, WHO Fellowship, and Best Paper Award in many places. He is also the life member of National Academy of Science, Indian Public Health Association, Indian Society for Medical Statistics, Indian Association of Occupational Health, and many more. He has also traveled abroad for research and meeting purposes. He has around 250 publications in the national and international level reputed journals. His thrust area is occupational health, environmental health, and public health. He is also the editor of 2 books entitled Environmental and Occupational Exposures, Reproductive Impairment, and Recent Trends in Environmental Health. He is also the reviewer of many national and international peer-reviewed journals. Apart from this, he has also guided 42 AFIH students and 26 MPH students for their dissertation and thesis work. He is visiting faculty to many organizations such as Public Health Foundation of India, Manipal University, Government of Gujarat, IGNOU, etc. We feel grateful to have you, sir, as our esteemed chief guest today. I now humbly invite Dr. Tiwari to enlighten us with the storehouse of his knowledge and experience. Sir, please.
Good morning, all. Respected Principal, Dr. Asha Agrawal ji, Vice Principal, Dr. Josna Galgale ji, Convener and co-convener of this conference, esteemed faculty joining through online mode, esteemed faculty sitting in this auditorium, and the online as well as offline delegates. First of all, I am grateful to the organizers for giving me this opportunity, though a last moment entrant, but then it is my pleasure and I'm really grateful because I am always fond of being among students, because that gives you a learning experience. Because when I started my career, I started from college as a lecturer, and now I am in a research organization. So I always feel that among students is always give you something new every day. So for that, I am really grateful to the organizers. I congratulate Sri Sathya Sai College for organizing this. Am I audible? So I congratulate the college for organizing this 2-day conference, and particularly the theme of the conference, because it is all encompassing. And as previously told by Principal Ma'am and the convener of this conference, that COVID has affected almost every dimension of life. So be it the social life, be it the economic life, or be it the life which the intellectuals live. So we all know that 2 years back, when it first came to our notice somewhere in February 2020, we were almost having no infrastructure to tackle that scenario. But then at the same time, if you see the challenges which was posed by COVID, it has also highlighted what our capabilities are and what our weaknesses are. Now, in the terms of, if you see the capabilities which were highlighted, we have developed vaccine in a record period of time. we have vaccinated the huge population of our country in a record period of time, and we have saved several lives. And all this credit goes to the science, technology, and innovation. And so the theme of the conference is very relevant for the current context. I'm proud that my organization, Indian Council of Medical Research, took a lead in controlling the situation in the country. Within 15 days time, we noticed first case in our country. We were ready with the guidelines which is to be followed. Later on, government during declaration of first lockdown implemented those guidelines through different state governments, and within 1 month period we were ready with the isolation of the virus at that time. So we were ready for the process of developing the vaccine for that. At the time of declaration of first lockdown, there was only one institute, that is National Institute of Virology, which was entrusted with all virus related research. But then within a short period of time, maybe 3 months, we had more than 1,000 testing laboratories across the country. The testing kits were a big challenge, and ICMR, through different institutes, we have a network of around 27 institutes across country, mandated for different purposes. So we are mandated for environmental health, occupational health, malaria research, tuberculosis research. But at the time of the crisis, all 27 institutes were mandated for the COVID related activities, and we fulfilled the responsibilities which were put to us. And it was the collective effort of various state government agencies, various central government agencies, various research organizations, that we successfully tackled COVID pandemic during last 2 years. When you compare it with most of the developed nations, say European Union or say USA, which has suffered a lot as comparison to India. But these were the dimmer side of the pandemic. But then it has also opened several opportunities and several challenges also. First challenge with respect to research, when we all were thinking, and that is a classical example, USA is a classical example where most of these research were targeted toward non-communicable disease, be it cardiac problem, be it cancer, be it other problem. But then this pandemic has shown that infectious diseases are here to stay. And more importantly, more and more we are going for environmental degradation. We have to suffer the consequences of that. There was about some saying about One Health when SARS-1 hit and somewhere in 2001. But then this pandemic has shown that it is high time that we should now think of One Health with respect to avoiding such future pandemics or epidemics. And the scenario is very evident. In the recent time, maybe in a period of 2 or 3 years, we had 3 or 4 scares, like it was Nipah, it was Zika, it was COVID, and now then monkeypox. So there was a scare of number of such viral diseases which has emerged. Hopefully or fortunately, we could not suffer much from other 3 except for COVID. But then it is high time that we should think of One Health approach. So the research has changed from infectious disease or non-communicable disease research to a comprehensive One Health research, where we have to see these infectious conditions from host to the reservoir to the environment and to the shedding in the environment. So this has changed. Again, COVID has forced that there should be social distancing or isolation. And so it has also emphasized the importance of technology in the health sciences. There were instances where machine learning or artificial intelligence was applicable in other fields of technology, but then health was almost untouched before COVID. But then this COVID has emphasized that these type of techniques are very much relevant with respect to the public health. And so even at ICMR, we are having consortium, we are inviting proposals on these aspects of technology merging with the health of the people. So these were some of the newer dimensions which were opened with respect to research. And no doubt, when AIDS came, we at ICMR opened institute for the research for that particular virus at Pune. And now again with this pandemic, frankly speaking, the One Health Institutes which was planned by ICMR, it was before pandemic, but then this pandemic has emphasized the role of this institute, and so ICMR is opening a National Institute of One Health at Nagpur. So that will be doing research which is related to all such type of instances or events which will be happening. So these are the newer dimensions which were open for the research in the biological sciences or the public health. With respect to therapeutics, again, we know that what happened during the second wave of the pandemic, that everybody was running from here to there for different modalities of the treatment. And this pandemic has shown that we still have a lot to improve with respect to viral infections, because many of these drugs which were being given to the sufferers of COVID infection were simply trial and error. So this has opened that we have to be doing more research with respect to the therapeutics part of viral diseases, more specific antiviral drugs. So these are new area with respect to therapeutic research or therapeutic innovation is concerned. With respect to innovation, again, it started from the larger cities or from the towns, and then it percolated into the remote villages of our country. And this was a real challenge, how to approach these remote villages, because we all know, though we are in the 75th year of our independence, and much development has happened in these 75 years, but still we lack quality healthcare infrastructure at the periphery level or at the rural level. And so it is a real challenge how to strengthen the health infrastructure in those remote areas, because we all know that many times these remote areas are crowded, and such type of infection, if it enters into these areas, it spreads fast. And you know that in subsequent versions of COVID virus, the infectivity was higher, though fortunately the pathogenicity was on the lower side. So even in those scenarios, some light was shown by ICMR, like we developed we utilized the drone facility to deliver vaccines into the remote villages of the northeastern state, say Manipur or Nagaland, where you cannot reach through either route, whether it's air route or road route. It takes time. So what we have done, we utilized the drone facility, which is a very good facility and which is being used for many purposes. So we have used that facility to deliver vaccine into those areas where you cannot reach so swiftly in time. So these are some of the innovative technologies which should be used. Even somebody said in the beginning that even education also, or for that purpose, interaction with people. So these technologies were there, like video conferencing or a WhatsApp call. So these technologies were there before pandemic also. But then this pandemic highlighted technology can be very useful in conveying the message, in dissemination of the knowledge, and at the same time saving the cost. Regarding entrepreneurship, again, when our Honorable Prime Minister said that this is a scenario where you can find so opportunity in the time of crisis. So, and again, he said that it is again time because across the globe, every country was disrupted. So business was largely disrupted because of lockdown, because of restrictions and all those things. So this is again a time when we go for vocal for local. So it is a time that we develop our inherent capabilities in different areas of business also, so that if such type of thing happen in future, we are self-reliant. And so came the Atmanirbhar Bharat scheme, where government has taken a lot of initiative to support our MSMEs so that they can become self-reliant. And again, through the governmental organizations like ours, we are regularly being oriented, compelled to follow that route of Atmanirbhar Bharat, which is very good for countries like ours. And so I feel that all the aspects of COVID, from sociology to economics to microbiology to pathology, if you see, though it has certain devastating effects on our life, but then at the same time, it has opened a Pandora of opportunities where we as intellectuals can contribute and can help our nation building those capacities. During pandemic period, maybe initial 1 year, ICMR has published more than 300 research papers, and it was a collective credit goes to all the scientists across 27 institutes only on COVID. So our then Director General, Dr. Balram Bhargav, he has released 2 volumes of such publication, and these publications were published in Lancet, in New England Journal, in Nature, and all those high reputed journals. Otherwise, if you see the before pandemic, if you have to publish your article in these type of reputed journal, you should have some co-author from the developed nation, maybe from US, maybe from UK or some other. So because it is usually the research at the Indian level, it is seen with some suspicion. But COVID has shown that we are capable of producing research of international quality. And so that the results were visible to everybody. And so with this, all these opportunities which are opened by the COVID, I feel that this organization of this conference is very timely and very relevant. And I'm very certain that with all the esteemed faculties across the globe, this conference will definitely be a learning feast for all the delegates who have joined this conference, either in person or through online mode. And I wish all the success to the organizers of this conference and to all the participants for their future. Thank you.
Thank you so much, sir, for your valuable insights. We all are definitely benefited out of it. What COVID has taught us is the best way to avoid war is to be prepared for it. What COVID has taught us to be self-reliant. And it also suggested that we need to look for the opportunities at the time of crisis. It taught us to be aware about the surroundings, social distancing, and areas of research. I'm recollecting few lines from Robert Frost, and miles to go before I sleep, and miles to go before I sleep, which suggests that we have a lot more to work, lot more to do before we go for the final sleep. That is, therefore, search, research, and re-research is the need of the hour. Thank you very much, sir, for your valuable insights. Finally, as an appreciation and acknowledging graceful presence of respected Dr. Rajnarayan Tiwari for the time and valuable inputs that he has shared with us today, I would like to invite our Principal, Dr. Asha Agrawal, to offer memento to our guests as a token of remembrance. With this, on behalf of our institution, we thank you for your time. Your presence has been valuable to us today, and we certainly look forward to have another interactive session with you in future too. Thank you so much, sir. We are honored that another eminent personality, Dr. Rajan Dubey, has joined us. Moving further in the academic and research-oriented interaction, it's a proud privilege to have another eminent scholar with us, Dr. Rajan Dubey, Chief Global Strategy at SRF Life Sciences, Healthcare Scientist Singapore, as our esteemed keynote addressee. We welcome you, sir. He has joined us online. Before inviting him for his deliberation, I take the opportunity to share his inspiring bio brief. May I request our guests to please take their seats.
Is that me? Am I audible? Am I supposed to speak now?
Yes, sir, you are audible, but Megha ma'am is going to say something about you.
All right.
Am I audible to you, sir? Dubey, sir?
Yes, ma'am.
Yes, Megha ma'am, please continue.
Dr. Rajan Dubey did his MBBS in 1999, PG Diploma in Preventive Medicine in 2002, MBA Health in 2006, and MBH in 2014. He has worked with various areas of research and dealt with many companies and agencies associated with public health management and development programs. He had worked with top pharmaceutical companies around the world and has 12 years plus experience in public health program management and implementation of large programs in various states of India. He had also led immunization programs with WHO in UP and UNICEF in Madhya Pradesh. He has also held donors and support programs in various states of India. He has held responsibility of state and national level trainer for routine immunization program, syndrome case management, home-based newborn care, and training modules for medical officers supported by WHO. He has an extensive experience in gender budgeting SOP for public health hospitals, technical paper evaluation, developing protocols for biomedical waste management for health and municipal facilities. He has also been associated with successful planning, funding implementation of key ethics for Bill Gates Foundation for RTI/STI services across the 3 states of India. He has also been the member of various advisory boards of health and nutrition program, research institutions, and guided and supported various research projects in India. He has also developed social daycare centers of CSWs in Bengaluru, Hyderabad, Mumbai in India. And the list is endless. He is presently working as Chief Global Strategy at SRS Life Sciences, Healthcare Scientist Singapore. And right now, he is joining us from Germany. I take the privilege to invite Dr. Dubey for his deliberation. Sir, please.
Thank you very much, ma'am. And I hope I'm audible enough and taking the cue from Dr. Tiwari, very interesting views that he has presented on how India took up the challenge of COVID and how vaccination has been done. So I'll just give you a brief of what I'm trying to show you, all students and all the dignitaries. I would not like to name and rename all of them, but my sincere thanks to all the organizers for this opportunity for the students. My brief session, I would like to show you the window in which the biotechnology right now is operating and how we at Singapore, I'm right now in one of the biggest, I would say, conference, or you can say the shows, in trade shows in pharmaceuticals and biotechnology in Germany. I'm just finishing it. So I'm just trying to show you a window in which you will be able to see how the world sees from the human medicine perspective. Later on, if you have any questions or any specific queries, I would be more than happy to entertain them later on. You can have my email later on, or you can just send me the queries and I would be happy to answer and have an interaction later on as well. Nonetheless, I'll try to share my presentation. If there is any difficulty, please let me know. Is that visible to all of you? Is that fair? My screen is visible, right? I assume. So this is my presentation. I am a consultant to various pharmaceutical companies as well as the government. So these are my opinions, and this is for educational purposes only. So my view to take on the pandemic is, now that it's 2 years old, a population of the size of Finland has so far died, and tens of millions more dealing with the side effects, and multiple more. And there are a number of cases where you can see that the number of deaths are much challenging, and who has died how much is a very debatable concern. But nonetheless, what I would like to point your vision is how the industry has seen as investment has poured into the various innovations across the world. And in 2021 alone, around $44 billion were raised. I'll try to show you a few of the graphs as also in which I'll show you how much it has risen from the previous ones. And I can show that in 2020, it was like almost like an acquisition of health and tech companies rose by 50%. You can see this 10 years of global health innovation funding. So guys, you are all young. The future is looking bright. You can see investment is coming in. And I'll also show you what are the areas in which it is coming in so that you can prepare yourself for this opportunity that is coming in, because I believe there is a synchronization between what the world wants, what the funding is there, and where the opportunities for the students will be. So behold, that might be of some interest to the new student. So you can see here in the graph, it is visible to you that starting from the digital care to the remote monitoring to insurance to surgical robotics, clinical decision support with AI, digital therapeutics, hospital operations. This is how the system has changed, and this is how the investment scenario has changed. If you can see, the most consistent ones were remote monitoring of the wearables and the drug development with AI. This is where most investment has come. Most hopes have come for the telemedicine that has not quite taken care of right now, or has not taken off in the appropriate scale right now. But I believe it will. nonetheless, these are the three most consistent areas, and then comes the insurance. So that is like a payer's view. That is much required to effectively implement the telemedicine. So rest of all, you can see are the areas in which it has been working. So nonetheless, let me give you some 8 or 9 windows in which most innovations are coming, and therefore the most funding is going on. You can see this very simple graph in which I can tell you, not a graph, I would say a picture, in which how the patient's journey has happened during the time and how people wish to be receiving the care at home, because this is where the most of the end result has lying it to, whether it's a post care of the patients or when the patients have been restricted to home or home quarantine has happened. This is how the care has to be given. And this is an area which is evolving very fast, because people are knowing that at the time when the surge happens or any sudden fluctuation in the number of patients happens, it is not the hospital infrastructure that can handle them. So they have to segregate. And this is the home care, and especially with the geriatrics, and we are entering into the phase 4 of the demographic cycle. This is what is going to be the mainstay of care. So, as Dr. Tiwari was saying, that SARS was there in 2003 and now COVID-19 in 2019. The response that the world has shown in terms of how they will respond to a pandemic started from 20 months now to less than 2 to 3 months. This is how BioNTech and Moderna have done some splendid jobs there. And China has developed its Sinovac. Russia has developed its own indigenous vaccine. So did we in some time. So nonetheless, we all responded to it with our might and hopefully did some dent. So just 1 second. So this is the global biotechnology market by region. As you can see, in 2015 to projected to 2026, you can see that the North America has a major share of pie, and the Europe, third is Asia Pacific. So we are in the scene, we are progressing, not that much, but nonetheless, investment and the market is growing in our regions as well, which is the Southeast Asia. Biotech fields that are being taken up: bioindustrial, bioagriculture, biopharmacy. So you can see that the biopharmacy is the one that is taking up the lead, much more stronger than the other, and especially after 2020, that has taken a big lead. So you are standing and you can see that all of them are progressing. You can choose any one of them as per your liking. So future interesting fields in medical technology, as I was telling that I would like to show you the window. One, I think, is the bioconvergence. This is an area where most interesting things you will be able to see in future, I believe. Data and digital are rapidly merging with the latest techniques in drug discovery, engineering, and biotech. This is the merger of bioconvergence. This will unlock the potential of personalized medicine. We can discuss the personalized or the precision medicine later on, but I would just like to point you out. You can see into bioconvergence as a field, and then you will be able to see how the new opportunities are come up. The only thing is, beyond anything, especially in a country like India, we must not caught up in endless planning. We have to get moving with clear structural steps that we can achieve this transformation through deep partnerships with aligned objectives between governments, providers, payers, and the technology companies. This is what that has to emerge to take the right opportunity and the right benefit from these opportunities that are coming up later. Technology has been always the heart of biotech. I always say, so the computer technology is the one that the big data, the big mining farms, you will see a lot of buzz in this area where people would be saying they want to make the best use of the data, whether it's a clinical trial data, whether this is a simple human data that is being recorded at the hospitals or the periphery or at the personal level by the wearable sensors and the wearable technologies that are coming up. This is the area that is going to show you the first window. So this is, behold, this is the first window I'm trying to show you. Computer technology that is going, computer companies, those you are seeing working in other fields, they will be venturing into this field very soon, or they are already doing it. Second is stakeholder collaboration. Stakeholder collaboration is going to be very important in this area. I believe that I'm joining you from Germany, and most of the time I live out of India, so my work usually is by virtual meetings. You can see that even the government has to rely, or even the people, those who are trying to display their information or trying to discuss the various research and coordinated research at this point of time, used very simple tool like you are using Zoom at this point of time, or Microsoft Meeting, or such kind of platforms. This actually is going to increase the stakeholders collaboration, which is going to shorten the timeline, and new ways will appear in this particular area. So this stakeholder collaboration is the one that you must master to see how you're going to take the approach of it. This is the second window. Clinical trials. This is going to be decentralized. This is going to be very different than what we have seen in the past. Clinical trials is one of the biggest area where most of the money of the pharmaceutical company or the biotechnology company actually goes. If a drug trial has to take place, it takes maximum number of time this time. You can see that the vaccines have been issued for public use within no time, with a very short time, only because decentralized clinical trial can be taken, the data can be collaborated, results can be interpreted and posed for usage. This is where this area is going to show the maximum expansion in terms of money, especially in the developed world. Why I'm saying developed world? Because there the maximum research on the drugs and other things takes place, including the antiviral ones, as Dr. Tiwari was saying. Clinical trial activity overall, I would like to show you how this area is, because I think this is an area of concern for us as well. So clinical trial activity has been sustained through the pandemic. Industry has adapted to disruption and developed new approaches to enable research to continue. Almost 5,500 new planned clinical trials have been started. You can see the clinical trial registry. It is 14% over 2020 and 19% higher than 2019. So you can see that it is consistently growing. The aggregate number of study participants are intended in new trials exceeded 2 million in 2021 for the first time. That means the number of patients, those who are recorded, so you can understand the variables, the data, the collected, actually increases tremendously. And therefore, you can see the linkages as I've shown to the first slide. The computer technology, the decentralized clinical trial, and the stakeholder collaboration all are merging into this activity. And you can extract the data from such large number of clinical trial and the registrants to these trials. Personalized treatment of rare diseases. This is one of the most important things that we are seeing the challenges with. You can see the genetic sequencing starting from 2.7 billion for the Human Genome Project. Now we are just about $100, I believe. That is how your genome can be mapped. We have not yet been able to harness the entire potential what we have taken out, but I believe in coming time we will be able. Yes, ma'am.
मिक्सी टाइप का कुछ चल रहा है?
Yes. Hello. Hello.
Sir, I have muted everybody. Now you please unmute. Rajan sir, please unmute yourself.
Okay. Thank you. Thank you very much.
Thank you, sir.
Somebody was speaking. Thank you very much. So this has enabled the development of personalized treatment plans and targeted therapies, which are more effective than less specific therapies because they focus on patients' genetic and molecular makeup. So you are going to see a lot of trend which is coming up in personalized treatment of rare diseases. You can, if you want to understand from me what is going on just in your country right now, you will see the top business leaders actually are removing their entire old workforce. Entire means entire. Multiple layoffs in Pfizer, in Novo Nordisk, in multiple companies are happening. They are only keeping the team that is going to work on rare genetic therapy, gene therapy drugs that are coming up. US FDA has lined up with almost 50 NDAs. So you can see a lot of development in this area where the people would be and the bigger companies would be lying for this opportunity first. Oncology, neurology, and infectious diseases. This is where the launches will come. Almost 169 oncology launches in the past decade, including some of the most groundbreaking research. Now you can see the monoclonal antibodies and none less than checkpoint inhibitors coming into place. So checkpoint inhibitors are the ones that are going to change the way we see this particular world and how we see into the oncology. There are multiple products in the pipeline, R&D pipeline, and Europe is declined over the past 15 years, but I believe US share has been stable, and therefore even Korea and China have taken a lot of advances into this. This is the areas you can see. This is the area where the clinical trials have been registered, COVID NAS launches. You can see COVID-19 was there, but nonetheless, oncology, as I was saying, 169 launches in the last year. So this is where the maximum number of products are coming in. These are the most important ones that have been taken up by the industry as a challenge, and that's why this is where the area is coming. This is the next window, value-based pricing models. That means this is most interesting, where the finance will merge with a new product. That means how you are going to be using the new technology, how you are going to be using a medical device, how you're going to use the technology that is coming up for personalized medicine, which would be very costly for some people, but at a larger level. This would be the most interesting task. In the environmental sciences, value-based pricing could be targeted to percent of waste cleaned through the use of microorganisms. This is the area that is coming up, but this is entirely new window that you will be able to see some experiments, some new innovations coming up. Next is monoclonal antibodies and biosimilars. We, as a company, are a manufacturer of monoclonal antibodies, and we in this world see these as leading the charge against various diseases, including infectious diseases. As you can see, even in COVID, you must have heard about tocilizumab, which is the IL-6 inhibitor, interleukin-6 inhibitor, and that is the one that has been used to stop or arrest the cytokine storm that has there. So one of the most effective drugs beside remdesivir. So this is where you can see quick development, but heavy cost, very costly medicines. Therefore, biosimilars will catch up in the future. This will be like the generic for the monoclonal antibodies. Nonetheless, this is the biggest area in the pharmaceuticals that is going and under development. Acceleration of innovation cycles. You need to understand that the innovation cycles that have taken the time and the toll of almost 5 to 7 years for a vaccine have started to come. You can see within a few months, things have taken place. So expect a very fast innovation cycle in the industry, and therefore multitude of, as you can connect to all the dots that I've been discussing here before with you, multiple stakeholders, computing technology, the new personalized medicine systems, and ability to conduct decentralized clinical trial. This is how the innovation cycles will be accelerated and time will be shortened. You can see that this is the most important to find out even the treatment for the rare disease disorders, because that is where the short timing will help most of the time. You can see here, phase 1, phase 2, phase 3 clinical trial, they all have their challenges. After phase 1 to phase 2, phase 2 to phase 3, you can see that this year, probably some of the biggest dropouts you'll be able to see after the phase 3, but the problem is probably most of the people have taken a bigger risk in 2021 that is going to pay off also, but yes, you will see a lot of rejections as well this year. So the composite success rate for the pipeline fell to a 10-year low in 2021. driven by drops in phase 1, phase 2, phase 3 success, while slightly offset by an increase in regulatory submission success rates. The regulatory submission means NDA applications have been accepted. And the phase 3 success rates saw the greatest drop in 2021, declining to 48%, which is 98% lower than the 10-year pre-pandemic period. I consider that to be both the mechanisms of action and increasing next generation and first-in-class pipeline preparation. This could be the, I would say, risky approach that they have taken, but that's all right. You have to do take some extra risk in these kind of times. Cell and gene therapies. This is the slowly growing field, which is very interesting. And we are closely watching it, and the world is closely watching it. You will hear a lot of interesting news, and it will look like a dream pipeline to all of you. But you will see some very, very interesting products here. And you must be hearing from the world that FDA has approved the first ever gene therapy, Chimera. You will see a lot of people now. It has been approved for the sickle cell anemia as well. So you will see a lot of interesting things that will be coming up and clinical trials, but behold for it. This is a new area where we are learning, but yes, this is the area which is going to expand in future. This is how it is going to be. All other biologics, 318 right now in the FDA approved cell and gene therapies. Cell therapy is 7 and gene therapy is just 2. Gene modified cell therapy is 5 and HPC cord blood is 8. So you can see this percentage is slow and short, but it is going to increase tremendously after some time. Private investment in life sciences, this is how it is coming up. Cell therapy, gene therapy, both are picking up. And you can see cell therapy is picking up now. It has, after 2019, it was lagging behind the gene therapy. Now it has picked up the pace, and most investment has come up in cell therapy. And so gene therapy is progressing consistently, but nonetheless, the cell therapy has taken lead now. The number of approved treatments remains extremely small. Gene therapy is approved by 14 cord blood based treatments have been approved. So you can see cell and gene, phase 1 is 11%, phase 2, 9, phase 3, 6, and filed is 2. So you can see that the small molecule chemistry, large molecule biologics, numbers are low, but nonetheless, you still have over 1,000 ongoing clinical trials. You'll see a lot of failures, but I'm saying that the window is opening up now. This is a graph which is very busy slide, I would say, and it is trying to show you various drug approvals and regulatory actions that have been taken place and the various areas that have been picked up, starting from oncology to CNS to COVID-19. This is a very busy slide. I don't think this can be discussed at the moment, but this will actually show you how different timelines have been used by the different, I would say, therapy areas, even during the COVID period. So I would say FDA was busy approving a lot of therapies. So vaccine development, the last one. Search for preventing the spread of COVID-19 has forced the scientific community to consider new ways of developing vaccine. Yes, it is. And it's going to be very challenging, interesting, controversial, information, misinformation, public health handling, a lot of debate is out there. You would be seeing a lot of debates in social media as well, but I would like to tell you one thing very clearly, is that whatever happened, nonetheless, we saw a development, and this is not going to stop, this is going to continue. You would be able to behold in the future how the vaccine will be developed, is benchmarks have been changed forever. So the world is more bullish. We are seeing with very hopeful eyes to tackle multitude of our diseases through vaccine now. So even you will hear that the pioneers of the mRNA vaccine are now working towards treating cancer also through vaccine, because that is now the new thing coming up. So you will hear a lot into vaccine development now. So pre-COVID vaccine trials, contracting and planning. You can see the changes that has happened. This is the Sightline Traveler, January 2022, FDA. This is how this has been, I would say this is the global trend in R&D. Overview, you can see that. Explanation is timelines were reduced by 70%. This is phenomenal. This is how the real timeline has been shown, and now we have leapfrogged through a different era. The time savings were enabled by regulatory and government investment, which de-risked the critical decision making, innovation, and resourcing, because the public money, when it comes to an area, the actual regulators and also the financiers become a bit more risky, and therefore new results come up. So you can see the result very well. 70% timeline reduction, which is very huge. Wide space efficiencies were driven by urgency, execution partner alignment, and leader engagement, leading at the least 2 months of saving from accelerated contracting and trial planning alone. And I know 2 months doesn't seem much, but they actually lead to huge time savings and money savings at our industry. Here I rest myself, and we can engage into a discussion, but since this is an hybrid mode, I don't think if there are any pertinent questions that I can take up at this point. Nonetheless, what I request students to see into these areas where you want to work, look into the future, into the industry, and this is going to give you a sort of snapshot in which you will be able to see how the industry is viewing it, how new opportunities will come, what are the areas in R&D that you will be able to harness for your own benefit, what are the areas your teachers can help you to go through and look into the new features and contribute to the society in a positive way. That's your motto. So I hope I have done justice to a few minutes, and if there is any questions I can take up, or as I've told, you can give me questions later and I would be more than happy to answer at any point of time. Please feel free to ask any question. I always accept brick batting also. So please don't hesitate. You can ask any question. You can offend me as you may like. I'm happy.
Thank you so much, sir, for your valuable insights. I feel that your lecture and your presentation was so self-sufficient that the participants here are feeling contented enough. But in future also, if we have questions, I will request Dr. Rupa Nandi to mail you with questions, sir. I hope we'll be getting the answers from your end, sir. Thank you so much. And what we could conclude, what I could conclude, I am not the person from the biosciences, but I was listening very carefully to it. And the presentation, what I could conclude is the slides and graphs that you presented, it gave an overview of the areas with the maximum potential. And the areas that you highlighted will definitely come not only to the interest of the investors, but also to the researchers as well. Your presentation also covered varied areas of biology as I was going through your presentation. Agriculture, veterinary, environmental sciences, and human medicines as such. And we hope your presentation will definitely help the researchers in the biological field to investigate the world of biotechnology in a more holistic way. And we have more challenges to overcome, but more opportunities to grab in the field of human medicine. Thank you so much, sir, for being here with us.
Thank you very much, ma'am. And I really appreciate this opportunity
so much, sir.
to interact with you. Thank you.
Yeah, thank you so much, sir. We are now at the end of inaugural session of 2 days international conference. Gratitude is greater than being great. Even the bees who sip nectar from flowers hum their thanks when they leave. We are blessed with the gracious presence of all the dignitaries and the wonderful participants. I now invite Mrs. Supriya Singh, Assistant Professor Chemistry, to present formal vote of thanks.
A warm and pleasing morning to one and all. On behalf of the entire organizing team International Conference, I'm glad to express my vote of thanks. First of all, I would like to express my heartfelt gratitude to our chief guest, Dr. Raj Narayan Tiwari, sir, Thank you, sir, for gracing this occasion. A big thanks to our keynote speaker, Dr. Rajan Dubey, sir, and of course, the central force behind all these activities, our chairman, Dr. Meena Pemplapure, ma'am, Principal Dr. Asha Garwal, ma'am, and Vice Principal Dr. Jyotsna Galgale, ma'am. I would also like to extend my word of thanks to gathered all dignitaries, faculty members, research scholars, and repeaters. A special thanks to technical staff. Last but not the least, I'm thankful to those who are working directly and indirectly to make this program a success. Thank you.
Thank you so much, Supriya. Sometimes challenges and struggles are what we need in our lives. May we welcome every effort, every struggle, every challenge, and open our wings to fly. And we aspire to achieve the objective in this 2 days conference. Once again, I thank you all for the wonderful participation. Yes, your presence makes a difference. We acknowledge the presence of all our participants and delegates, both who have joined us online and offline, and want to capture the moment as a part of our journey of learning. I therefore request all of you to assemble for a group photograph near stage, after which we'll have a short tea break and we'll assemble again. All the participants, please come gather near the stage for a group photograph. Thank you, everyone. Participants are requested to assemble here after 10 minutes break for the next session. Thank you.
Tea break is over now. Varsha ma'am, are you ready? Varsha ma'am, you are requested to unmute yourself.
Good morning, distinguished participants, ladies and gentlemen, and my dear friends. Hi, Dr. Varsha Saxena. I'm pleased to welcome you to first technical session of this 2 days international conference on post-COVID trends of biological sciences towards research, therapeutic, innovation, and entrepreneurship. In the time of COVID-19 pandemic, it has become blindingly obvious that science, technology, and innovation are indispensable for recovery from the crisis. We are witnessing rapid advances in synthetic biology and information technology leading to innovation. What is important is deployment of these innovations on a scale that can reach the target audience. Together, let's dive into exchange of ideas. On this pleasant morning, it's my privilege to welcome our chairperson for the session, Dr. Renu Mishra ma'am, HOD Botany and Microbiology, Sri Sathya Sai College for Women, Bhopal. Ma'am, may I request you to please come on the dais? I would now request our co-chairperson for the session, Dr. Renu Srivastav ma'am, Assistant Professor Zoology, to please come on the stage. As is our tradition, let's welcome our esteemed guest with a sapling. I invite Dr. Rupa Guhanandi, convener of the conference, to welcome Dr. Renu Mishra ma'am. Thank you, Rupa ma'am. May I call upon Dr. Sunita Yadav, co-convener of the conference, to welcome Dr. Renu Srivastav ma'am. Thank you, Sunita ma'am. I now request all the dignitaries to take their seats. Today, I have the honor of introducing Dr. Renu Mishra ma'am, our chairperson. Ma'am is an internationally recognized educational visionary. She believes in empowerment through education and has left no stone unturned for the progress of girls and young women. Dr. Mishra is currently employed as Associate Professor, Head Department of Botany and Microbiology in Sri Sathya Sai College for Women, Bhopal. She is also IQAC Coordinator for the college. She completed her post-graduation from Barkatullah University, Bhopal in 1985. She then obtained her PhD degree in 1990 from Barkatullah University. Her research interests lie in the areas of aeromycology, allergy, soil microbiology, medicinal plants, and pharmacology. Dr. Renu Mishra has guided 14 students for PhD and more than 100 undergrads and postgrads for dissertation. She has published many chapters in books and one book which is recently published in Springer. The title of the book is Applied Mycology. She has completed 2 UGC-sponsored projects and published and presented more than 65 papers in various national and international journals and conferences. She has been the proud recipient of many awards, which include 3 Best Paper Awards, Bharat Gaurav Award, Best Microbiology Teacher Award, Senior Scientist Award, and Career Excellence Award. She is well known for her leadership and dynamic vision. We welcome you, ma'am, here. Our co-chairperson for the session, Dr. Renu Srivastav ma'am, is working as Assistant Professor, Department of Zoology and Biotechnology, Sri Sathya Sai College for Women. She is member Board of Studies in Zoology at Barkatullah University. She is guiding 5 students for their thesis work and has published more than 25 research papers in various national and international journals. 1 book and many chapters in books are also credited to her impressive work. In her various capacities as assistant professor, researcher, member of many committees, She has always inspired us with the benchmark she has set. We are honored to have you here, ma'am. With your permission, may I start the session? Before calling upon my first participant, let me just tell you that each participant will get total 10 minutes of presentation. So 8 minutes will be the presentation time and 2 minutes will be question answer session. Our timekeeper, Dr. Nishi Yadav ma'am, will remind you at 7 minutes so that you can conclude your presentation in 1 minute. So I request the participants to kindly stick to time limit. I call upon my first participant, Muskan Dhakar. She is from Sathya College for Women, and her topic of presentation is Impact of COVID-19 Pandemic on Healthcare System, a Review.
Om Shri Sai Ram. A very warm good afternoon, all of you. I am Muskan, and the topic on which I am presenting my review paper was Impact of Post-COVID on Healthcare System, a review. In history, for the first time, a health crisis had shut down the entire global economy, painfully demonstrating how inseparable healthcare and the economy has become. Across the globe, healthcare systems were not designed to deal with this crisis. Uncertain, large-scale health challenge has required urgent mobilization of resources and affect the whole population. In this review article, what I am explaining or what I try to explain is that what are the conditions of healthcare system before pandemic, that is in 2019, at the moment of pandemic, 2020, 2021, and after the post-pandemic. Now moving to the next part, what is actually COVID-19? It is an acute respiratory illness in humans caused by coronavirus, capable of producing severe symptoms and in some cases death, especially in older people and those with underlying health conditions. It was originally identified in China in 2019 in Wuhan. We all are very much familiar with the symptoms of coronavirus, that is fever, shortness of breath, cough, etc. In India, the spread of COVID-19 was initially characterized by the lower case numbers and fewer deaths compared with numbers in many different countries. The virus has already created havoc in global markets. It remarkably impacted the sourcing, procurement, and the management of necessary medical equipment inventories. And due to this, there is huge shortage of oxygen supply, hand sanitizers, treatment facilities like ventilators, beds, etc. Now moving to the next part of my presentation, that what are the traditional healthcare systems in India? Originally, the country employed a strategy focused on the containment of the virus, applying measures such as quarantine individuals. People are starting quarantine, they are starting isolating themselves in their homes, contact tracing, and restricting the travel from people from areas where caseloads were high. Over the years, healthcare system in India has been largely centralized, expensive, and impersonal, such as care is inaccessible, unresponsive, and unaffordable. Since we are not mentally and financially prepared before the pandemic, so it impacts global economy in large number. COVID-19 pandemic exposed these realities further. It has also made it urgent to redesign the system that is more decentralized, distributed, and responsive. What are the healthcare system of India and how it has impacted COVID-19? So India's healthcare sector faced a stern test as they struggle with lot of shortage of beds, oxygen during the 2 waves of COVID-19. Since the second wave exposed the short of oxygen, India produced more than 7,000 tons, but the problem lay in transportation and storage. The mental health of India's healthcare workers were severely impacted. Now, case study on USA health facilities. What measurements or what measures the USA health facilities have taken to prepare for the surge in hospitalized patients with COVID-19? Hospitals needed to create more negative pressure rooms, hire a backup workforce, educate staff, obtain PPE, that is personal protective equipments, and PPE shortages. All non-urgent and electrical surgeries procedures were cancelled to free up the essential hospital staff and hospital beds. Now in China, so what are the measures that Chinese government took place during the pandemic? After the SARS epidemic, the Chinese government quickly launched an overall healthcare system to improve access to such things as services and high quality supplies. It invested in system for disease surveillance and reporting as well as epidemic prevention and control. Centers for disease control were built across the country. These are the availability of hospital beds by country per 1,000 people. Now, case study on the healthcare in Singapore. The country was one of the earliest to report COVID-19. Initially, was second to China in total cases. It established the National Center for Infectious Diseases and 900 response to public health preparedness clinics. The most important factor that shaped Singapore's response to the pandemic has been its vaccination campaign. Booster vaccine doses were engaged and were determined as necessary be considered fully vaccinated. Even prepared the public for a possibility of taking booster shots periodically to maintain their fully vaccinated status according to the vaccinated differentiated measures. These are the impact on healthcare system. The medical devices or the equipment, diagnostic, pharmaceuticals, health insurance, healthcare facilities, health information technology. Now, what are the healthcare reforms? Or we can say that what is the transformation after post-COVID? So first is recruit, train, and prioritize healthcare workers. Since healthcare workers are at the heart of any strong healthcare system, throughout the COVID-19 pandemic, frontline workers, which includes community health workers, have selflessly risen to the challenge. That means providing them with necessary training, making sure they are first in line for vaccines, so they are protected, and financially and emotionally supporting them. This support is needed not just so that healthcare workers are equipped to do their job properly, but to ensure the profession is an appealing one. Second is establish effective surveillance and response system. So the routine use of big data and artificial intelligence approaches to model crisis and to identify and understand the weaknesses of existing system would be necessary in order to strengthen the existing structure. Development of communication technology-based approaches. Obviously, the transformation in healthcare would not be possible if it is not associated with a technological innovation in communication, machine learning, and transportation. The expansion of Medicare healthcare coverage amidst the pandemic is a major step in the right direction, as well as increased delivery of the healthcare closer to home for chronic neurological patients. Development of financial models to support the cooperation and crisis preparedness. I hope that we all are aware of this app, that is Arogya Setu app. And with the utilization of self-help apps, a future-branded approach is likely to emerge, where e-mental health solutions occupy a greater part of routine services. Additionally, currently, expertise can be used in expanding a wider public mental health approach. Include COVID-19 vaccines in routine immunization packages. What are the transformative approaches? Create lean organization by removing redundancy, re-engineering processes using technology, and offering remote care. Transforming the process of research and development based on the gained experience, encourage research and innovation. Now, the conclusion, that is, the COVID-19 outbreak serve as a reminder that proactive planning for healthcare emergencies as well as intensified commitment to global public health preparedness remains necessary. The realization that technologically empowered solutions can be implemented and work should constitute the benchmarks for the greater integration of such technologies as a part of routine healthcare design and provision. Optimal outcomes can be attained where both patients and healthcare providers become active participants in this process. Thank you.
Any questions from the participants or audience?
What are the new government policies regarding the healthcare after the COVID? Ma'am, government has initiated some funding platforms for the people who are not
cannot afford the healthcare facilities like ventilators. Even the rich are having difficulty in admitted in hospitals. I didn't recognize the name specific. Any other question from audience? Okay, thank you, ma'am.
Thank you, Muskan. Our next participant is Yashomi Borasi. She is from Sathya Sai College for Women, and her topic is effectiveness of COVID vaccines. Om Shri Sai Ram. Om Shri Sai Ram. Good afternoon to everyone present here, one and all teachers and all the students. I am Yashomi Borasi from Sri Sathya Sai College. I am going to represent a presentation on topic effectiveness of COVID-19 vaccine, a review. As we all know that in the end of 2000, as we all know that in the end of 2019, a new disease recognized such as severe acute respiratory syndrome, SARS. The disease was caused by an unknown SARS coronavirus 2 that is characterized by high infectivity among humans, as well as the different adverse effects that can cause to the individuals. So, to fight against this virus, the vaccines has been developed with the effectiveness of each vaccines. For introduction, what is vaccine effectiveness? It is a measure of how well vaccination protect people against outcomes such as infection, symptomatic illness, hospitalizations, and death. Vaccine effectiveness is typically measured through observational studies specifically designed to estimate individual protections from vaccination under real-world conditions. This graph shows the vaccine effectiveness in each countries with each vaccines developed by them. These are the charts which show the vaccine effectiveness, which is agree or disagree, means which pass the approvals or not. There are many vaccines which are developed with scientifically researches, like this shows efficacies of each vaccines, how they work on immune systems on the human body, like effectiveness in adolescents and children have effectiveness rate more than in the elders, more than the older persons as compared to the smaller ones. COVID-19 vaccines continue to protect against SARS diseases. And what does it mean if a vaccines offer 80% effectiveness of it? Like here it shows that the persons who are vaccinated are more vaccines show more effectiveness on it, and the person who got non-vaccinated person are having the higher risk of viral infections against it. How effective are COVID-19 vaccines developed by each country? The graph shows that the Pfizer's and BioNTech's, which is developed by US and Germany, have the efficacy rate of more than 95%. And then it is, second one is Moderna, which is mRNA-based vaccines having the efficacy rate of 85%. Then ground COVID Sputnik vaccines, which is developed by Russia, having the efficacy rate of 90%. Next is Novavax, which is developed by USA, having the efficacy rate of more than 80%.
Then the Indian vaccines are Covishield, Covaxins, which are approved in India for the trial and also got the effectiveness, also show the effectiveness more than 80%. And also Sputnik was also approved for the trial and had successfully trial more than 80% accuracy rate of it. Then here it shows that the vaccinated person having the less chance of infection than the person who are unvaccinated. These are the effectiveness shot, which show the effectiveness of each vaccines under placebo group in vaccinated group. The positive result will show the side effects of COVID-19 vaccines having the severe headaches, higher fever, red spots of hemorrhaging under the skin, heaviness in the chest, trouble breathing, body-wide rashes, vomiting more than 5 times, or fecal palsy, muscular weakness, seizure, fainting. All these conclude that the vaccines which are developed are useful rather than non-vaccinated persons who are not vaccinated through it. They have more chances of getting infections than the vaccinated persons. So this also conclude that the doubt on the persons who are having that the vaccines are vaccines having less chance of survival of that also exclude that vaccine which is work more than vaccine which work more than 80% chances having the accuracy rate of more survival than unvaccinated persons. And the safety and effectiveness have safety of the person who get effectiveness who get COVID vaccine have the efficacy rate of more than 90%. Here it is also showing that the person who got vaccinated have the safety purpose. First, safety patronized. Second, tried and tested. Third, highly effective. This show that the safety prioritized. We have confidence that COVID-19 vaccine, including those by Pfizer or Moderna, will be critical tool in the pandemic. No steps were skips in the development of process, and safety was a top priority. Second, tried and tested. More than 74,000 people took the vaccine during clinical. Thank you. Any question? This should be the group vaccines used in India. Yes. Yes. Two vaccines which are used in our India, Covishield and Covaxin. Which one is the best and which has less side effects? Now, Covishield has accuracy rate of 90%, which shows that the person having vaccinated by Covishield have less chance of getting infection than the Covaxins and lesser chance of side effect than Covaxins. Yes, ma'am. Yes, ma'am. Any other question?
Yashumi, am I audible to you? Hello.
Yes, ma'am.
Can you tell me, do you think that alone vaccination can prevent COVID-19 infections?
Ma'am, please repeat question.
क्या वैक्सीनेशन अकेला कोविड-19 इन्फेक्शन को कम कर सकता है?
No, ma'am. There are major things we have to also take. Not only vaccination will against this virus, but we have to take the protocols against it. Like we have to social distancing is one of the best example for it, which can also reduce the chances of getting infections against it. Also hygiene.
Okay. ठीक है. Thank you.
Any other?
One question, Beta.
Yes, ma'am.
Is there any difference between the first dose and second dose of the vaccines that we have taken and the booster dose, which is being taken after 6 months or 3 months or 90 days? Is there any difference or are they the
Ma'am,
same?
there are various variants to this coronavirus like Omicron and Delta variants, which are more stronger than the normal coronavirus. That's why the booster dose is announced to stimulate the immunity of humans. So third dose is also announced for that.
And now, but is it mandatory? Is there any difference in the composition of it? Because
Ma'am,
people
it
are
will-
reluctant to take that dose.
Ma'am, it will boost the immunity more than first and second dose.
There is no difference. These doses are just required to boost us. Right? Thank you.
Thank you, ma'am.
Thank you, Yashami. Our next participants are Poonam Baghel and Anushri Nair. Poonam Baghel and Anushri Nair.
Good afternoon to everyone present here. I am Anushri Nair.
And I am Poonam Baghel.
And we are here to present our review on the medical importance of Indian gooseberry on post COVID-19 treatment. Coronavirus disease is an infectious disease caused by the SARS-CoV-2 virus. It is the most dreaded pandemic of our time, which led to a state of chaos in the mightiest nations of the world. The immune system plays a significant role in the response to any foreign organism, be it bacteria or virus. The COVID-19 virus can spread from an infected person's mouth or nose in small liquid particles. These particles range from large respiratory droplets to smaller aerosols. COVID-19 affects different people in different ways. Most infected people in different ways because of how different our immune systems are. Most infected people will develop mild to moderate illness and recover without hospitalization, whereas few of us may it may be fatal for few of us. Most common symptoms include fever, cough, tiredness, loss of taste or smell, and serious symptoms may include difficulty breathing or shortness of breath, loss of speech or mobility, or confusion and chest pain. A key objective in long-term COVID management is to identify and proactively manage complications and support patients through the recovery phase with the goal of preserving their health status. It is necessary to formulate proper treatment protocol for these patients to help them restore physical and respiratory function and to reduce anxiety and depression, particularly in patients with multiple chronic conditions, to restore a good quality of life. Although the Chinese system of medicine has been explored, there is still more to be done in exploring solutions from time-tested Indian systems of medicines like Ayurveda. Ayurveda has enough potential and possibilities both for the prevention and treatment of long-term COVID. Clinical trials carried out in the past 1 year on the management of COVID-19 has reported that Ayurveda management is beneficial in mild to moderate cases of COVID-19 without long-term lingering symptoms. This is the reason why, after recovering from acute phase of disease, many patients seek for Ayurveda treatment for long-term lingering side effects. Ayurveda treatment, when taken along with modern medicine in acute stage, then it can reduce side effects and there will be no long-term residual symptoms. Here is the Bentham and Hooker classification of Phyllanthus embolica. Ayurveda believes in the healing properties of medicinal plants, one of which is amla. Amla is a well-known medicinal plant that is also called as Indian gooseberry, Emblica officinalis. It is a medium-sized deciduous tree, typically 8 to 18 meters in height, belonging to the Euphorbiaceae family, and is a native to tropical Southeast Asia, particularly in central and southern India, Pakistan, Bangladesh, Sri Lanka, southern China, the Massachusetts Islands, and Malaysia. Generally, trading of amla tree is done largely from wild extraction in south parts of India. However, several cultivations are grown in commercial plantations in Uttar Pradesh, Maharashtra, Gujarat, Rajasthan, and Andhra Pradesh. Amla is not only rich in natural vitamin C, but also contains a wide variety of bioflavonoids, which further enhance the effects of natural vitamin C and often purported to have considerable health benefits. Vitamin C is found particularly in citrus fruits and green vegetables. It is essential in maintaining healthy connective tissues and is also thought to act as an antioxidant. Its severe deficiency causes scurvy. It is an antioxidant that helps prevent cell damage caused by free radicals. Vitamin C is the best natural immunity booster and is one of the strongest antioxidants we can obtain directly from nature. But why so much emphasis on amla when the world over, general citrus sources like oranges and lemon are commonly consumed? For the same purpose, the most impressive fact about amla is that it has close to 8 times more vitamin C than an orange and 17 times more vitamin C than pomegranate. The amla is not only rich in natural vitamin C, but also contains a wide variety of bioflavonoids, which further enhance the effects of natural vitamin C. The bioflavonoids also play a role in controlling infections and healing wounds, and it's a powerful antioxidant that can neutralize harmful free radicals. Some examples include polyphenols, gallic acid, and oleic acid. But vitamin C mainly helps make several hormones and chemical messengers in the brain and nerves. It is essential for a normal and well-functioning post-defense mechanism, and its pharmacological application is believed to enhance immune function.
Not only fruits in its raw form, but several commercial products are also available that make use of amla's constituents, which are Chyawanprash, Triphala Churn, Brahma Rasayan, Madhumega Churn. These products also contain medicinal properties like anti-scorbutic, diuretic, antibiotic, laxative, and anti-dysentery. So Filambilin and is an antioxidant obtained from fruit amla pulp has been found to have mild depressant-like actions on the central nervous system. Amla and vitamin C juices are also considered as a good liver tonic. There is also a good demand from the industrial sector for preparing various healthcare products like hair oil, dye, shampoo, face creams, and tooth powder. So after learning the benefits of amla, we have here proposed a small methodology for extraction of different metabolites of the fruit. Number one is collection of amla fruits, which is done manually using bamboo ladders, harvesters, or by shaking free such that the fruit is collected on the ground where the plastic and canvas sheet is spread. Number two is surface sterilization. The fruit is then thoroughly washed under the cold water and then dried. Extraction of pulp of fruit by removing the seeds. Process involves the sorting, washing, de-stoning, and fine pulping. And the filtering of extract can be done through the filter paper or a muslin cloth, and juice then taken out into a clean container. And the last is identification of phytochemical constituents. These can be evaluated by different chemical methods and cellular methods. Thus, we conclude in short, the COVID-19 crisis not only challenges disease control and crisis management, but may also have long-term and far-reaching impacts on state, societies, and international cooperations. COVID-19 caused by SARS-CoV-2 virus can cause mild severe respiratory illnesses, including death. Most infected people will develop mild to moderate illness and recover without hospitalization. Ayurveda believes in healing process of medicinal plants, one of which is amla.
The fruit amla is rich in beneficial metabolites, antioxidants, polyphenols, and bioflavonoids, which often purported to have health benefits. Thank you.
Any question?
Thank you.
I just want to ask you, while preparing for this presentation,
Yes.
did you come across any reference or any paper where post-COVID, any research has been reported that gooseberry has got its effectivity against COVID-19?
Ma'am, we have come across, sorry. Ma'am, we have come across many sources from which we have collected many evidences that amla has many health benefits and is also a booster of immunity. Because COVID-C because COVID-C leads to weakened immune system. Amla has several properties like Amla has vitamin C, which boosts the immune system.
No, I got your point.
Yes, ma'am.
This is well accepted that amla is an immunobooster.
Yes, ma'am.
And immunoboosters, they help to boost your immunity and thus help you to fight against the virus.
Yes, ma'am.
But what I want to know, after 2020, did you get along any publication where this has been proven by tested results that amla has been found to be effective against coronavirus? I'm simply asking, did you come across?
Ma'am, many research papers have found the potential of amla, but none have proven them as such.
Yes. Thank you.
I have one question.
Yes, ma'am.
According to you, which is the main phytoconstituent of amla, which is responsible for boosting immunity?
Ma'am, it surely has to be vitamin C, but other bioflavonoids also, along with vitamin C, help in the boosting of immune system and lead to recovery from post-COVID symptoms.
What are these phytoconstituents?
Phytoconstituent include polyphenols, ellagic acid. I'm sorry if I don't know if I'm pronouncing it right. Ellagic acid and gallic acid. Yes.
Thank you. Any other questions? Thank you, Anishri.
Thank you.
Thank you, Anushri and Poonam. It was a wonderful presentation. Our next participant is Ritu Sharma. She is from CSIR AMPRI and her topic is Impact of COVID-19 on Global Agriculture.
Ritu. I think we should need it to pronounce. Sairam everyone, my name is Ritu Sharma and I am going to present a review article on impact of COVID-19 on global agriculture. Track we are going to follow through upcoming slides. So a pneumonia-like disease was found to occur in Wuhan district of China
in Wuhan district of China in early December, and within 2 months, it captured whole global scenario. It was transmitting on a very fast rate, and the attributes leading to the fast transmission of COVID-19 were the viral structure, of course, the high risk of interhuman transmission, long incubation time, its zoonotic nature, existence of asymptomatic carriers. COVID-19 has led us to a very devastating impact. Total 1 to 1 million people were found to be contaminated by COVID-19, and it has led to the death of 2.3 million people. But another sector which was very badly hit by COVID-19 was global agriculture. So first we need to analyze what is the global agricultural scenario. From figure 7, I wanted to understand you, like, for example, we Indians are importers of fertilizers from China, largest importers of fertilizers from China, and then we produce lots of crops such as wheat, jute, barley, so many, and then we are going to sell it to other countries, for example, USA, Turkey, etc. And this is how a chain is initiated, a global agroeconomic chain. Now, what happened during COVID? This chain was broken because of the lockdowns imposed. Lockdowns and quarantine imposed, impacting the fertility of farmers, global transportation, and industries shut down. This has brought down the economic loss and eventually leading us to a state of global food insecurity. So this can be cleared by figure 9. So from this paper, I need to drag your attention towards our need for preparedness for another worst situation which might happen next year ahead. So for dealing with the matter of food security, we need plants which are more yielding, climate resistance, drought resistance, which are more nutritional, which are pest resistance, and who have better shelf life. This can be achieved by role of biotechnology, by implementing techniques such as gene manipulation, hybridization, transgenesis, and genetically modified organisms. Though there are some concerns in employment of biotechnology in plant and agriculture. These concerns are allergy, food toxicity, animal refusing to eat these kind of crops, threatening the pool of genetic diversity, and also this method is very costly. But we need to understand that there is a need to cope up with the scenario, to understand that we have a huge population that is growing on and on. So we need to develop a balance. We don't need to choose between environment and development and technology. We just need to choose somewhere in between. So we need to rely on these measures, choose something in between, and develop global corporation, local governments for aiding literacy to the farmers, for upgradation of these technologies. We need we need regulatory policies regarding GMOs, and we need proper GMO field test, and also we need to strengthen the biosecurity measures for checking the entry of new pathogens in the cycle. Thank you.
To my understanding, the main reason for the impact of COVID on failure of agriculture that we have highlighted is unavailability of fertilizers to the farmers.
Yes, ma'am.
Fail the chain of transportation. I want to ask you, at the time of pandemic, our PM proposed Atmanirbhar Bharat Yojana. Okay, so can you somehow relate this to the farming practices? So that during post-COVID period, farmers, they have developed some new techniques or some new alternatives to avoid such kind of difficulties in future.
Can you please repeat the question?
Simply better, can you suggest कुछ ऐसे provisions या कुछ ऐसे ideas कि आत्मनिर्भर योजना में lot of startups have come up, and people they are using their new ideas to fight the situation. And आत्मनिर्भर योजना जो है वो initially इंडिया को independent बनाने के लिए काम किया. तो क्या फार्मिंग सेक्टर पे ऐसा कुछ हमें या स्टार्टअप या आइडियाज मिले हैं कि फ्यूचर में इस तरह की आने वाली मुझे डिफिकल्टीज को हम फाइट कर सकें?
ओके मैम, ऐसा कोई स्टार्टअप इंडिविजुअली तो नहीं है फार्मिंग सेक्टर में. हाउएवर एक भारत फर्टिलाइजर करके भारत खाद करके या फर्टिलाइजर करके ऐसा कुछ तो है कि गवर्नमेंट ने पूरा एक डोमेन किया है कि हर फर्टिलाइजर उसी ब्रांड नेम के अंदर बिकेगा. और बाकी अभी मोस्ट रिसेंट में जीएमओ क्रॉप्स को जो है जीएमओ मस्टर्ड भी अभी इंट्रोड्यूस होने वाला है. होपफुली अगर सेंट्रल गवर्नमेंट ने इसको मंजूरी दे दी तो या बीटी कॉटन के बाद ये मस्टर्ड ही आएगा. और मैम ऐसे तो बहुत सारी रिसर्च होती रहती है बायो पेस्टिसाइड्स पे, बायो फर्टिलाइजर्स पे. और इसके अलावा जो हमारे पास दिल्ली में एक प्रॉब्लम है जो कि एयर पॉल्यूशन से, स्टबल बर्निंग से, उसमें एक ICAR की रिपोर्ट के मुताबिक एक स्टबल बर्निंग के लिए पूसा करके कुछ जो है डिकम्पोजर एस्टैब्लिश हुआ था. तो मुझे इतना ही याद है अभी. इसके अलावा तो मुझे अभी कुछ शायद याद नहीं हो.
I have one question. Is genetically modified crop can increase the yield?
Yes, ma'am. It can increase if we want them to. See, when we are talking about genetically modified plants, we are manipulating their genes. We are putting in the genes that we need to enhance their characters. So if we want them to increase their yield, तो हम उसमें वही वाला जीन इंड्यूस करेंगे, वही वाला जीन एक्सप्रेस कराएंगे और फिर वो इंक्रीज हो जाएगी यील्ड. अगर हमें पेस्ट रेजिस्टेंस कराना है तो हम पेस्ट रेजिस्टेंस का कराएंगे. तो इस तरीके से हम उसकी यील्ड भी बढ़ा सकते हैं जीएमओ प्लांट की. और ऐसा हुआ भी है. आप बीटी कॉटन का एग्जाम्पल ले सकते हैं मैम. जब पेस्ट रेजिस्टेंस कम हुआ तो यील्ड ऑटोमेटिकली बढ़ गई थी.
But any other side effect?
मैम मैंने स्लाइड्स में भी आपको बताए थे बहुत सारे साइड इफेक्ट्स हैं इस चीज के. एलर्जी कॉज होती है. और क्या होता है कि जब हम जीएमओ प्लांट को इंट्रोड्यूस करते हैं मतलब एक फील्ड पर, तब क्या होता है जानवर उसे खाता नहीं है. और साथ में क्या होता है कि जो एक जीन पूल होता है पूरे मतलब कोई भी जैसे ह्यूमन है तो ह्यूमन का भी एक जीन पूल है, कोई प्लांट है तो प्लांट का भी कोई जीन पूल होगा। तो ऐसे में क्या होता है जब हम कोई नया जीन इंट्रोड्यूस कर देते हैं किसी भी जीएमओ में, कोई भी ऑर्गेनिज्म में, तो क्या होता है कि एक सुपर वीड बनने की संभावना हो जाती है। उसे कोई एनिमल नहीं खाएगा तो वो ऑब्वियसली बढ़ता जाएगा, ऐसा हमने पढ़ा भी हुआ है और इसका दूसरे एग्जाम्पल्स भी हैं काफी। तो मुझे लगता है कि बहुत सारे कंसर्न्स हैं इसको।
Any other question?
No more questions. Varsha ma'am, please continue.
Ritu, does it affect the soil fertility?
सॉइल फर्टिलिटी को अफेक्ट करता है ये जो
मैम
जो भी है
जी मैम, मैं समझ गई, मैं आंसर सोच रही थी क्योंकि मुझे ऐसा लगता है कि करना तो नहीं चाहिए पर करना भी चाहिए। मैं कंफ्यूज हूँ, मैं रीजन आपको बता सकती हूँ। देखिए, करना इसलिए चाहिए क्योंकि यील्ड जब वो ज्यादा देगा तो उसको जो है ज्यादा न्यूट्रिशन की जरूरत पड़ेगी और सॉइल फर्टिलिटी जो है वो ज्यादा इनहेंस मतलब ज्यादा सॉइल से जो है न्यूट्रिशन डिराइव करेगा। और नहीं करना चाहिए इसलिए क्योंकि मुझे ऐसा लगता है कि जब हम उसे फर्टिलाइजर्स तो प्रोवाइड नहीं कर रहे हैं तो उसे हम उसे फर्टिलाइजर्स तो प्रोवाइड नहीं कर रहे हैं तो फर्टिलाइजर्स से जो फर्टिलिटी स्पॉइल होती है वो नहीं होगा। मैम हाँ, नाइट्रोजन फिक्सेशन जीन करेंगे तो फिर तो वो फर्टिलिटी को अच्छा बढ़ाएगा ही, लेकिन जनरली के दो सिनेरियोस बता रही थी आपको।
Okay, thank you, Ritu. Thank you.
Thank you everyone.
Thank you, Ritu. Our next participant is Anjan Yadav from Global School of Excellence.
Om Sri Sai Ram, respected judges, teachers, and all my dear friends. I, Anjan Yadav from the Global School of Excellence, working as a TGT teacher. I am going to present my topic, that is post-COVID complications, long-term effects of coronavirus after recovery. We all are concerned with the present scenario, that how COVID affected, what are the consequences we are facing, what are the problems we are facing during the COVID period. But what about the consequences or the problems that we are facing after the COVID? There are some topics that I relay my attention. It is a review paper. So since December 2019, we all are aware with the COVID-19. It is being caused by the SARS-CoV-2 virus, and it is the infection which is rapidly swept up the whole world. And as the data from the February 9, 2021, 107 million confirmed cases are there and 2.3 million deaths worldwide. So what are these post-COVID-19 conditions? So when we are talking about this, they are the post-acute COVID-19 syndromes, PACS, which are affecting the 30% of the patient who are coming in contact with. But when we are talking about it, so 1 in 10 people testing positive of COVID-19 exhibit these symptoms more than or longer for the 12 weeks. So when we are seeing this long COVID in around 10 to 15% of the people, they are having the COVID-19 symptoms. It is not necessary that they are being hospitalized in the hospitals. But these scenarios maybe come from the patient who are neither facing the COVID, but still they are facing this long-term symptoms in their body. So being tested negative, but still they have the symptoms like shortness of breath, anxiety, chest pain, cough, allergy, rashes, and body ache. Many more symptoms are there which people are facing nowadays. So what are these long COVID symptoms? They are the lingers which remains in your body, means for the week or a month. So according to the National Institute of Health and Care Intelligence, these are the signs and symptoms that develop during the infection, or it may longer in your body for more than 12 weeks. And this, if we are going to discuss on this, so this can be the acute COVID-19 symptoms which are in our body up to the 4 weeks. And if the same symptoms are persisting in your body for 4 to 12 weeks, so they are the ongoing symptomatic COVID-19 condition. But when they are longer for more than 12 weeks, and we can't say any explained diagnosis will be there to determine or to identify such consequences why they are happening in our body, so they are termed as the post-COVID syndromes. So how these long COVID or post-COVID syndromes exist in our body and what they are doing in our body? So three emergencies theories came in our mind, and the scientists, those who have done the researches, they came in the way. The virus either remain in your body as a reservoir, like in the gut, and whenever it is getting the chance, it is being expressed and it is showing the symptoms in your body. Secondly, the part of the virus maybe remain in your bloodstream, either the protein structure, and whenever it is getting the suitable condition, it is being expressed in your body. Third one, virus can set up the autoimmune conditions in your body. Whenever it is getting the chance, it is overreacting, and even through the virus can no longer be in your body, but still you are facing the symptoms that are being symptoms which are similar to the COVID patients. So from the epidemiology and the pathophysiology, the study has demonstrated that SARS-CoV-2 and other coronaviruses are using the angiotensin converting enzyme 2, ACE2 protein for the cell entry. Means they are using this mechanism to enter in our body. This ACE2 protein is the integral membrane protein which is useful for our body. So they are using this pathway to enter in our body, in our organs, and subsequently they are harming our organs and leading to the long-term symptoms. So this ACE2 is having the important role in our body. It is a lung protection protein in our body, but when virus is binding with such a protein, it is causing the viral pathogenicity in our body. So this ACE2 can cause many organ damages. It can damage our heart, lungs, pancreas, kidney, many organs, and it can lead to the subsequent damage to that organs. If the virus is entering in our body, it can lead to the autoimmune responses. Our body is self-reacting to our similar cells, and it may be persistent virus remain in our body. It can lead to the organ damage, or it can lead to the microclot in our body. So when we are talking about the long COVID, so general symptoms may be the fatigue, pain, or fever. And if we are talking about the long-term syndromes or symptoms, so it can be of many types that we will discuss. So first, if we are talking about the brain or nervous system, so the symptoms may be headache, dizziness, strokes, seizures, and the long-term effects may be as soon as it will lead to the neurodegenerative disorders, Alzheimer's or Parkinson's, but there lack of the supporting evidence, we can suggest that this is the main root cause of these diseases. And for the eyes, it may lead to conjunctive diseases, abnormalities, and its long-term effects may be that the patient can experience severe oxygen depression, and it may lead to retinal and optic nerve damage. And if we are talking about the lungs, so how it is damaging the lungs? So it can be the cough, pneumonia, or respiratory failure. Its long-term cases will be, it can lead to the post-COVID fibrosis, which may be, further patient may have to transplant the organs, or it may have to lead to the many surgeries to get recover from the such symptoms. And if we are talking about the nose and mouth, so it may lead to the runny nose, and many more symptoms are there. And it may be the main problem we all are facing during the COVID and after the COVID is the loss of smell and taste. So this is the main problem we all are facing, and we are unable to identify that why we are facing such a situation after when we are not a COVID patient and after being recovered. So it may be the long-term post-COVID symptom that we all are facing. Then if we are talking about the liver, so it may leads to the liver dysfunction and severe injuries. And many researches have been done to find the evidence, but still no such evidences are found. And heart diseases also, many heart failure cases are being identified, but no such evidence are found. So we all are facing this long COVID symptoms in our body. Many patients are facing, many are recovered, many are being dead, but still it needs we don't have no treatment without research. So no treatment is there for such the symptoms. Only we can do the supportive care and we can take the preventive precaution for this. So what we can do, we can take the vaccinations because 85% of the record says that if we are being vaccinated, so there is a chance of reducing such COVID infection in our body. And 50% of the researches has been proved that if you are the vaccine, so these chances infection will not be occurring in your body. That's all. Thank you.
Nice presentation. Any question?
One question from my side. What is this angiotensin?
Ma'am, angiotensin is the protein receptor molecules which is present in our body. They are the receptors for the protein intake. And when
How could you identify? You have done research on it?
no, so ma'am, this is the review paper. I have just done the review and no research I have done. But in the future, if I will,
Molecular structure of angiotensin.
ma'am, that I didn't. I
You
just
have come to a conference of international level.
yes, ma'am.
That should be there.
Okay, ma'am. I will work on this.
Any other question?
Nice presentation.
Thank you.
Very nice presentation.
Thank you, Anjan. Our next participant is Jyotna Singh Chandwatchi. She is from Sathya Sai College and her topic is medical herbs helpful after COVID. There is some technical problem with her pen drive. Can we call upon the next participant, ma'am? Bhavna Kurmi, are you ready? Bhavna. Bhavna is our next participant and she's from Sathya Sai College for Women. Her presentation topic is nosocomial infection of COVID-19. That's right.
Please switch on your video camera.
Good afternoon, faculty and all my friends. I am Bhavna Kurmi. I am going to present a presentation on the review article, and my topic name is risk of nosocomial infection within the patient who survived after COVID. This is index. Where is the point I will describe in my presentation. First of all, I want to describe what is nosocomial infection. Nosocomial infection is when disease transfer one person to another person in hospital environment is called nosocomial infection. It is also hospital acquired infection. ऐसा नहीं है कि हम हॉस्पिटल में गए और तुरंत कुछ सिम्टम्स दिखाई दे तो हम उसे नोसोकोमियल इन्फेक्शन मानेंगे. जब कोई पेशेंट हॉस्पिटल में जाता है, उसके डिस्चार्ज होने के 48 आवर्स के अंदर कोई सिम्टम्स दिखाई देता है तो उसे हम नोसोकोमियल इन्फेक्शन कहेंगे. Classification types of nosocomial infection. First, central line associated bloodstream infection caused by Staphylococcus aureus. Second, urinary tract infection caused by E. coli. Third, surgical site infection caused by Enterococcus. Fourth, ventilator associated pneumonia caused by Staphylococcus pneumonia. And five, pathogen nosocomial infection like bacteria, virus, fungus. And six, environmental nosocomial infection. जब हम हॉस्पिटल में जाते हैं तो वहां पर बहुत सारे माइक्रोऑर्गेनिज्म और कीटाणु पाए जाते हैं, जिससे नोसोकोमियल इन्फेक्शन के चांसेस वहां पर बहुत ज्यादा बढ़ जाते हैं. फैक्टर विच अफेक्ट नोसोकोमियल इन्फेक्शन इज प्रेजेंस ऑफ माइक्रोऑर्गेनिज्म. सेकंड, पेशेंट का इम्यून सिस्टम. थर्ड, ट्रांसमिशन ऑफ पैथोजन. जब हॉस्पिटल साइड में अगर जब इक्विपमेंट यूज होते हैं, अगर वो सैनिटाइज और प्रॉपर क्लियर है तो जहां पर नोसोकोमियल इन्फेक्शन के चांसेस बहुत कम हो जाते हैं. लेकिन अगर किसी पेशेंट का इम्यून सिस्टम वीक है तो वहां पर लेस अमाउंट में भी अगर माइक्रो ऑर्गेनिज्म प्रेजेंट है तो वो नोसोकोमियल इन्फेक्शन के चांसेस बढ़ जाएंगे और वो नोसोकोमियल इन्फेक्शन से इन्फेक्ट हो जाएगा. नाउ COVID-19. Coronavirus is effect on respiratory system is called coronavirus disease. Now, कोरोना वायरस के बारे में तो आप सभी जानते हैं. कोरोना वायरस एक ऐसा पैंडेमिक है जिसकी वजह से सारे वर्ल्ड को बहुत सी प्रॉब्लम का फेस करना पड़ा. नाउ आई वांट टू डिस्क्राइब जो पेशेंट कोरोना से डेथ हो गई उसका रीजन. फर्स्ट, सबसे फर्स्ट उनका इम्यून सिस्टम वीक था. सेकेंड, उनमें हेल्पर सेल मीन्स बी सेल एंड टी सेल कम थे, जिसकी वजह से उनका एंटीबॉडी उस एंटीजन के खिलाफ काउंटरफेट करने में अनकेपेबल रहा, जिसकी वजह से उनकी डेथ हो गई. सेकंड, जो पर्सन कोविड से रिकवर हो गए उनका रीजन था, उनका सबसे बड़ा रीजन था कि उनका इम्यून सिस्टम बहुत ही स्ट्रॉन्ग था. नाउ द मेन टॉपिक इज जो पेशेंट कोविड से रिकवर हो गए उनमें नोसोकोमियल इन्फेक्शन के चांसेस क्या हैं. जो पेशेंट कोविड से रिकवर हो गए हैं, अकॉर्डिंग टू साइंटिफिक स्टडी 90% पीपल्स का इम्यून सिस्टम पहले से ही स्ट्रॉन्ग था. उनमें एंटीजन एंटीबॉडी इंटरेक्शन बहुत ही स्ट्रॉन्ग था, जिससे उनकी एंटीबॉडी एंटीजन पर डोमिनेट थी, जिसकी वजह से वो एंटीबॉडी उस एंटीजन को काउंटरफेट करके इजीली उसको डिस्ट्रॉय कर सकी, जिसके वजह से वो पेशेंट रिकवर हो सका. सेकेंड, पेशेंट जो कोविड से रिकवर हो गया उसमें बी एंड टी जो सेल्स थी वो इंक्रीज हो गई. अकॉर्डिंग टू साइंटिफिक रिसर्च, जब उन पेशेंट में 6 मंथ बाद एनालिसिस किया गया तो उनके बॉडी में बी एंड टी सेल्स में कोई भी गिरावट नहीं देखी गई. मींस इससे प्रूव होता है कि वो बी एंड टी सेल्स की संख्या बढ़ने से उनका इम्यून सिस्टम पहले से और भी ज्यादा बूस्ट हो गया. जो पेशेंट कोविड से रिकवर हो गए उनमें एक प्रोटीन डेवलप हो गया जिससे उनकी एंटीबॉडी और भी बढ़ गई, जिससे अगर वो पैथोजन फिर से उस पेशेंट के अंदर एंटर करता है तो वो एंटीबॉडी उसके अगेंस्ट इजिली काउंटर अफेक्ट करके उसे डिस्ट्रॉय कर देगी, जिससे वो पेशेंट इजिली रिकवर हो सकता है। सिंस द फाइनल एज, जो पेशेंट का इम्यून सिस्टम जो मतलब बहुत ही ज्यादा स्ट्रॉन्ग होता है वो इजिली उस कोविड से रिकवर हो सकता है, एंड बी एंटी सेल की संख्या बढ़ने से उसका इम्यून सिस्टम पहले से भी ज्यादा बूस्ट हो गया। सिंस द फाइनल एज, रिस्क ऑफ नोसोकोमियल इन्फेक्शन इज reduced. Thank you so much.
Thank you, Bhavna. Any question? आपने कैसे सैंपल्स कलेक्ट किए?
मैम ये तो एक रिव्यू आर्टिकल है। यस मैम।
Thank you, Bhavna. Our next participant is Jyotsna.
सोर से कुछ नहीं हुआ सकते सब थोड़ी दूसरी बात क्या
Good afternoon, everyone. Myself Jyotsna. Good afternoon, everyone. Myself Jyotsna Singh. Today I am here to present my review paper presentation on the topic of medicinal herbs helpful to promote recovery from the post-COVID. So, introduction of COVID is COVID-19 is a contagious disease and it's caused by SARS-CoV virus, and which starts infection by using the surface structure. There is a protein which is known as a spike protein present on the surface of SARS-CoV, and which bind to the target host cells and will cause infection. Introduction of medicinal herbs. Medicinal plants are also known as medicinal herbs, and medicinal plants can be any plant which has one or more of its part contain substances such as secondary metabaloids and which contain alkaloids, flavonoid and terpenoid, which is used for the therapeutic purpose and or which is a precursor for the synthesis of useful drugs. that is used as a medicines. And there are some common medicinal herbs such as one tulsi, nilgiri, sham tulsi, amla, garlic, aloe vera, ashwagandha. There are some researcher who says that there are some compound derived which is disrupt the attachment of SARS-CoV to the host cells. कुछ स्पाइक प्रोटीन जो प्रेजेंट रहते हैं सार्स कोविड के ऊपर, वो उनको मेडिसिन हर्ब्स उनका डिटैचमेंट करवाने में हेल्प करती है विद द होस्ट सेल्स. Common medicinal herbs are garlic, amla, aloe vera, ginger, turmeric, and tulsi. How medicinal herbs are helpful to promote the recovery within the patient after COVID. Medicinal herbs helpful to recovery from the patient after COVID is by boosting the immunity within patient, also before, during, and after the COVID. Boost of immunity will make the immune system strong and it's capable to fight back against the COVID. Example is Vernonia amygdalin. It's a bitter leaf, common name. The aqueous extract of this enhancing the immune response by increasing the level of white blood cells in the blood. There are some Indian herbs which help to boost up the immunity even after the post-COVID. First is neem. Botanical name is Antarctica indica, and it's an immunity booster, and it has some property, antiviral, antibacterial, and antifungal properties. It also helps to purify the blood by flushing away the toxins from the body, and through this, it can strengthen the immunity. Moringa oleifera, it's a botanical name of moringa. It contains vitamin C, which is the chief source which is a chief nutrient to build up the immunity. And there are some vital nutrients present also, which strengthen the cell, muscles, and tissue, and also help to repair the body. And a high level of some other substances also present. High level of potassium, iron, and calcium and amino acids are also present in the body to strengthen the immunity. And Triphala botanical name is Phyllanthus umbilica. Triphala contain antioxidative property of three fruits: Hartika, Bhimvartika, and Amla. And it is loaded with vitamin C and vitamin A both. Fourth is ginger. Ginger officinale is botanical name of ginger. It is a good source for preventing the respiratory tract infection, and it contains antioxidant, which is gingerol, and which help to power up the immune system and help to kill the viruses. So there are, during the first wave, COVID-19, what are the most used herbs? And on the basis of percentage, I have mentioned ginger, garlic, amla, this used, and 80% and 70% clove, lemon, and 60% neem, dalchini, basil, ajwain, and 40% is methi and kutki. And during the second wave, which herb is used the most, 80% amla, dalchini, garlic, and 70% neem, turmeric, pepper, and 60% basil, ashwagandha, and 50% methi and kutki. Benefits from the herbal medicines are they are very low in cost and prevent from viral infection and low toxicity or even no side effects. Conclusion. So here we hereby conclude that herbal medicines after COVID help to recovery the patient by increasing the immunity, and even after COVID also it contains wide variety of secondary metabolites, alkaloid, terpenoids, and very phenolic compounds, and these secondary metabolites help to inhibit the action of SARS-CoV. And there are some references. Thank you.
Joseph, can you tell us what is the role of secondary metabolites in plants?
Ma'am, secondary metabolites inhibit secondary metabolites में alkaloid, terpenoids और phenolic compounds होते हैं जो inhibit करते हैं SARS-CoV के ऊपर जो spike protein रहता है उसके action को inhibit करते हैं.
Dance. Plants have skin. We're exploiting them. We're exploiting plants to extract.
I'm sorry.
Okay, take time. Okay. My second question is, can you explain us how antioxidants are affecting something fibers? See, you are telling them that there are so many material plants, they have antioxidants in them. Can you explain the role of antioxidants in general?
They contain antioxidant property like amla contain vitamin C, through which they can inhibit the.
Okay. And what is the composition of Tripla? You have written only Phyllanthus amplica.
मैम it's a three fruits से वो मिलके बना हुआ है. Exact. आंवला एंड हरती की और एक और था, एक भीम, हाँ एक भीम बटिकी करके, आंवला और एक भीम बटिकी करके, उनका एक्चुअल बोटानिकल नेम नहीं पता है.
Okay, any other question?
Terpenes are present. Terpenoids are present in the plants. Various plants contain terpenoids as three terpenes and 3 carbon compound, which is present in various plants. And alkaloids are various alkaloids contain alkaline, which is various type of substances it's contain plants. Thank you, Jyotsna. Our last participant is Fiza Sayyed from Sathya College for Women, and her topic is impact of COVID-19 pandemic on scientific research in life sciences.
Good afternoon to one and all present here. Myself Fiza Sayyed from MSc First Sem. Today I am presenting a review on impact of COVID-19 pandemic on scientific research of life science. COVID-19 has affected all our lives in different manner. For some appears to be like messy, where as for some industries, it was like nightmare of their lives. But research industries were at the center of pandemic as the research is going on for how to get rid of the virus. Each and every trying their best to find the solution for this virus. The impact of the virus on the research enterprise itself is something being closely examined. In this paper, we will be discussing about the impact of COVID-19 research, that too especially on life sciences. Coronavirus. Coronavirus is a disease which is caused by SARS-CoV-2. Its genetic material is single-stranded RNA of 30 kilobase genome size. Its morphology is the surface of this virus is covered with viral protein spike, membrane, and envelope. Its size is similar to SARS-CoV with viral size ranging from 50 nanometer. Its common symptoms are fever, cough, tiredness, difficulty in breathing, and muscle pains. Its morphology is this virus has have a diameter of 50 to 200 nanometers. It has 4 structural proteins which plays main role, which has the first is spike, second is envelope, Third is membrane, which detains the protein. And fourth is nucleocapsid. What is COVID-19? COVID-19 is a disease it is a disease, infectious disease caused by SARS-CoV-2 virus. In some cases, the person might face difficulty in breathing or shortness of breath, loss of speech or mobility, or confusion, and many times chest pain. Those who are underlying medical conditions like cardiovascular disease, diabetes, chronic respiratory disease, or cancer are more likely to develop the symptoms of COVID-19 and get affected through this. Coronavirus can be spread from person to person and it is diagnosed with PCR tests. Old and young kids are more prone to this disease. Now, what is life science? The field of science that involves the scientific study of living organisms such as microbes, plants, animals, and human beings, as well as other living beings. While biology remains the centerpiece of the life sciences, technological advances in molecular biology and biotechnology have led to a burgeoning of specializations and new interdisciplinary fields. The extremely high research and development cost coupled with little revenue in the initial year of development and many life sciences firms. Here is impact of COVID-19 on scientific research. There are two impacts, positive and also negative. The positive impacts are number of laboratories and investigators have increased from a single scientific institute to 27 institutes in India. Large amount of fund is allocated in the research. Rapid increase in sharing of research findings. The health and healthcare was brought back to the forefront of societal issues. Negative impact, 57% of life scientists lost their jobs. Also, loss of jobs leads to financial consequences. 25% of respondents report 1 to 6 months of laboratories shutdown. Now, impact of COVID-19 on life science. The pandemic has accelerated some shifts in care delivery and life sciences globally. Introduced to the new pathogens with the help of science, and immediately the detection tests were and work on therapeutics had begun. Focus on infrastructure investments of life sciences. Now, impact of COVID-19 on life scientists. There are 77% of the respondents stated that their institute has been fully shut down. Essential services staff present on site. 90% reported a partial shutdown institute, and the remaining reported a basically fully operation. 57% of life scientists reported that they had lost some of their work. 25% of respondents reported at least 1 month and up to 6 months due to laboratory shutdown. And dry lab scientists were more likely to continue carrying out their work from home of dry lab scientists, but only 10% of wet lab scientists reported at least 80% per month. We observe some differences between male and female scientists, with males reporting fewer productive hours. This is in part due to the higher rate of females among wet lab scientists, and likely also reflects differences in children duties. Conclusion. Through this presentation, the aim is to show that research articles listing COVID subjects compared to non-COVID are partially marginalized. In particular, we look at several indicators of research production in life sciences. Number of published publications, number of impact weight publication, open number of publications related to clinical trials, number of application on grants, number of grant lists that existed before the pandemic. Our goal is to show that previously existing terms that match term related to each other, sharp increase in the number of articles. By showing this growth is strongly correlated with the relatedness of the mesh, which show that captures an important aspect of the growth dynamics and highlight the spike in very similar terms. Our emphasis is on knowing the importance of research, as it is a strong solution for saving lives and stabilizing economics. Hence, more and more research centers should be inaugurated. Thank you.
type of research center you want now on? What type of research center?
Molecular, ma'am.
Molecular biology.
Yes, ma'am. Yes, ma'am. Thank you.
Do you think that during pandemic, research in life sciences suffered?
Ma'am, can you repeat, please?
Do you think during the pandemic, research in life sciences suffered?
Yes, ma'am.
You sure, sir? Just now?
Yes, ma'am.
Your paper is.
Because laboratories are shut down, so they affected. Yes, ma'am.
वहां से निकलकर वहां में जो हुआ that was a laboratory only ना.
Yes, ma'am.
And it has affected other laboratories.
Yes, ma'am.
बंद हो जाओ हम आ गए.
Yes, ma'am. Yes, ma'am. दोनों मतलब पॉजिटिव भी नेगेटिव भी है एंड नेगेटिव इफेक्ट ऑल्सो.
Initially, it has suffered a lot.
Yes.
Okay. Any other question? Thank you.
Thank you, ma'am.
Thank you, Filza. With this, we have come to the end of our first technical session. All the presentations were wonderful, and it was so heartening to see the young minds engage in research in exchange of research ideas. May I now request our chairperson, Dr. Renu Mishra, ma'am, to please sum up the session.
Thank you, Varsha, for the nice introduction. Now, I would also like to express my gratitude to all the organizers for putting together such an illustrious session and for giving me the honor of chairing the session. The subject of the conference is of great significance to the scientific community and to the people in general. As the multiple waves of COVID keep infecting greater and greater numbers, we have all been very close to the impact that it has had on the health of the patient, both in short term and long term. The new term that has emerged in the context called the COVID syndrome need to be explored in greater detail. In that respect, I believe that this conference has been a noteworthy endeavor. Today, we had the opportunity to attend many informative papers presentation by the students both UG and PG courses. I would congratulate you all for your performance. In the first presentation, Muskan Dhakar spoke about the impact of the pandemic on the healthcare system. She highlighted how the unprecedented health crisis has resulted in the shutdown of the entire global healthcare system and impacted the global economy. It brought into focus the inability of existing healthcare infrastructure to battle the influx of the large number of the patient. The second presenter was Yashomi. She highlighted the efficacy of the available COVID vaccine in her presentation. She discussed the importance of vaccine in prevention of disease spread amongst the population. Further, she compared the efficacy of Covaxin and Covishield. She also elaborated on other vaccines in the market. The next presenter was Poonam Baghel and Anushri Nair. They explored on the medicinal importance of Phyllanthus amblica in post-COVID treatment. In addition to explaining the qualities of Phyllanthus, they adequately explained the use of Ayurveda in the post-COVID treatment. The next presenter was Ritu Sharma. She explained the impact of COVID on in global agriculture. She highlighted the massive impact of COVID
on the productivity of crops, safety, transportation, and supply chain management of foodstuff. She also pointed to the significant impact of the biotechnology can have on agri-system in the near future. Anjan Yadav has presented a review about side effects of patients affected with COVID during pandemic and discussed about the long-term impact of COVID-19 on different organ systems, which is very important for the management of system. Now, the Bhavna Kurmi presented her paper on nosocomial infection of COVID-19, which is a new challenge for healthcare professionals. She very well explained the different nosocomial infections and what are the chances of nosocomial infection in COVID recovered patients. Jyotsna Singh Chandravanshi highlighted the importance of medicinal plants to improve the immunity after COVID-19. She explained about some common plants and their medicinal values. And the final presentation was given by Ms. Fiza. She discussed about the impact of the pandemic on research prospects in the field of life sciences. She spoke about how the pandemic expanded the research efforts into new organisms, their structure and their impact on the creation of vaccines. These presentations were not only interesting and thorough, but they were presented in a very impressive manner as well. I would like to congratulate all the participants for their efforts and hard work in performance. I would like to end by again extending my thanks and congratulations to the organizer for hosting such an insightful and important conference and for giving me the honor of being a part of it. I wish you all the best. Thank you.
Thank you, Renu ma'am, for highlighting the efforts of our participants so beautifully. I would now request Renu ma'am, Dr. Renu Mishra ma'am and Dr. Renu Srivastav ma'am to please come on stage. Our chairperson and co-chairperson, please come on stage. I request our convener, Dr. Rupa Guhanandi ma'am, to present our esteemed chairperson and co-chairperson with certificates of recognition. Thank you, Rupa ma'am. And thank you so much, Renu ma'am. So here we end our first technical session. Let's break for lunch and we'll meet again after 1 hour for second technical session. Thank you.
Smita ma'am.
इसमें इनका वो भी है चार्जर।
Smita नहीं
Om Sai Ram. A very hearty good afternoon to all the dignitaries, academicians, participants, and students. I, Dr. Smita Nair, Assistant Professor, Mathematics, take this opportunity to once again welcome you all to the post-lunch session of the International Conference on Post-COVID Trends of Biological Sciences towards Research, Therapeutic, Innovation, and Entrepreneurship, organized by Department of Zoology and Biotechnology, Sri Sathya Sai College for Women, Bhopal. Giving pace to the session, it is a matter of privilege that we have with us Professor Dr. Jaroslava Swag-Gejic as our invited speaker. I extend my heartiest welcome to the madam. Professor Gejic will be sharing with us her wisdom on the topic subcritical water as an innovation tool in research. Before inviting her for talk, I would like to present a brief introduction of our speaker. Dr. Jaroslava Gejic is a full professor at the Faculty of Technology, University of Novi Sad, Serbia. She holds the doctorate on technical sciences. Her areas of research include subcritical water extraction, green extraction techniques, valorization of biowaste, chemical and biological characterization, and development of functional food and cosmetic products, to name a few. Her current research focuses on utilization of sub and supercritical water for exploitation of natural resources. Professor Jaroslava has designed and manufactured several technical equipments also. Her research work is valorized through her publication of more than 300 scientific books and research papers in international high-ranked journals. Dr. Gejic has been a visiting faculty at many institutes such as Faculty of Science, Department of Chemistry, University of Balearic Island, La Palma, Spain, Department of Biophysics, Prince of Songlaka University, Thailand. In addition, she has carried out numerous research fellowships at foreign institutes and universities such as Kelstad University, Bangkok, Thailand, Central Institute of Fisheries, Technology, Cochin, Kerala, Department of Chemical Engineering, Prince of Songlaka University, Thailand, to name a few. We are extremely honored to have such a magnificent personality among us. I now invite Professor Jaroslava for her talk on subcritical water as innovation tool in research.
Good afternoon to everyone. Thank you for this really kind introduction. And it is my extreme pleasure to be here today with you and to present a very interesting topic. I hope you'll find it useful.
Good afternoon.
So, I will present a very cutting-edge technology which addresses actually key words in your title of event conference, that is biotechnological sciences, research, innovation, of course, because I'll try to present how innovative this technology is and how it can be applied in different fields of research, and indeed for obtaining extracts with really high bioactivity. Thus, we're also addressing this therapeutic aspect. I will present subcritical water. Well, the use of this exceptional fluid, it's a cutting-edge technology which can be applied in very different, diverse fields. And of course, first of all, a few words about the state of this fluid, because the technology is based on pure water. Of course, it is well-known fact that every fluid has this phase diagram and critical point. So the great advantage of this technology is because it relies on pure water, environmentally friendly, safe, easily accessible, and compatible with cosmeticals, pharmaceuticals, etc. So for water, critical point is relatively high, 374 degrees. And pressure is also high, around 218 bars. I want to point out the differences in use of this hot compressed fluid in supercritical region and subcritical region, since this technology has totally different application in different fields of research when using in supercritical region. So if we are manipulating with this fluid, which I won't call water because it changes dramatically all properties, solvating, reactive, physicochemical, and all others. In supercritical region, which obviously requires very rigorous conditions, this fluid becomes extremely reactive. It is so reactive that even provokes corrosion of stainless steel. So hot compressed water under these conditions can be used for different purposes, and one of them is biofuel production from virtually any waste. But also my recent interest is to exploit this fluid and its reactivity for control the composition of different natural matrices and to valorize bio-waste by employing this high reactivity of water, which I explain a little bit later. However, if we are using this fluid, hot compressed fluid, in subcritical region, this solvent becomes competitive solvent to conventionally used organic solvents, but with green character. It is saved because we're using pure water by altering its physical, chemical, and solvating properties. So in this region, the idea is to avoid reactivity, which obviously increases dramatically with temperature increase. So in supercritical region, it's not anymore water, but the mechanism of the biodegradation follow radical mechanism. Whereas in subcritical region, the key mechanism is the increased dissociation of water, increase in concentration of hydroxide and hydronium ions, potentiating hydrolytical reactions. So we want to avoid reactivity in this region because we are using it as a solvent. As a solvent that replaces organic solvents. So we are able to produce different bioactive aqueous fractions with safety, environmental consideration, and of course, these fractions, which according to previous studies really demonstrated superior bioactivity, exceptional chemical composition, are compatible with products in many industries. I already mentioned food, pharmaceutical, cosmeticals. I would like to explain what kind of solvent water becomes when it is heated, because heated and compressed, but compressed to pressures which do not necessarily correspond to water vapor pressure at certain temperature. The idea is to manipulate polarity of water and solvating properties of this fluid. Namely, with heating, polarity of water drops substantially. So it becomes very flexible solvent, flexible and selective to different classes of chemical compounds. So we can finely tune polarity of pure water to target specific class of phytochemicals or bioactive compounds from different matrices, different samples, plant origin, animal origin, maintaining these exceptional advantages that we're actually using green solvent, safe solvent, and solvent easily accessible. So with temperature increase, once temperature is a key parameter, water polarity drops and it becomes even capable of solvating totally unpolar compounds, such as here at this slide, we have example of solubility of benzoepyrene, which belongs to polycyclic aromatic hydrocarbons, and it is highly hydrophobic molecule. So here we see remarkable increase in solubility in pure water of this highly unpolar compound. And maybe I should point out the differences because the researchers, of course, are very familiar with extraction of different classes of compounds with supercritical carbon dioxide, which has its application. But this technology of extraction, it is practically being at the opposite end of applicability of polarity, namely with supercritical carbon dioxide, we can manipulate with its polarity within the region of the electric constant from 1 to 2, meaning we are targeting unpolar compounds. Or alternatively, we can add modifier to slightly increase unpolar supercritical carbon dioxide. So I would like to point out to the audience that this technology, when used for extraction and for isolation of bioactive compounds, has certain advantages. That is high flexibility in terms of polarity range. We can manipulate polarity of water within the region from the electric constant 13 to 80 in critical point. So we can selectively target specific chemical class of compounds for the extraction. Of course, when we talk about extraction, it is also very important selectivity towards interferences. So, by increasing water temperature, we actually achieve polarity of conventionally used organic solvents such as acetonitrile, ethanol, methanol, which are toxic, which are not environmentally friendly. Ethanol maybe can be considered as generally safe, but there are also regulations concerning solvent residues in final extracts, final products. So actually water circumvents all these requirements because it's safe. I also want to mention that solubility of compounds which are already soluble in ambient water at the ambient temperature does not increase so dramatically as solubility of highly unpolar compounds, as we saw on the example for benzoepyrene, which is unpolar totally. So solubility of compounds that are moderately soluble in ambient water increases in less extent. For example, narrow, a common constituent of many essential oils, of many plants, increases only about 25-fold when increasing water temperature from 25 to 200 degrees. During this presentation, I will try to describe different streams of using this cutting-edge technology, in which I'm totally in love, because you will see in the end what can be done with pure, green, environmentally safe solvent. Innovation, of course, is, I would say, a key point, and in line, of course, with the topic of this interesting and interesting and very multifaceted conference I'm glad to take part. So my research is mostly conducted in a house-made extractor reactor, because this technology can be used for the extraction, but also for the composition reactions, depending, as I explained, on the temperature. And the composition reactions are conducted for different purposes, mostly for the production of platform molecules from biowaste, such as also organic acids, phenolic compounds, amino acids, to actually valorize biowaste, to avoid costly management of waste, to increase the value of biowaste, multiple times. So until recently and still today, commercial reactors, extractors based on subcritical water are not so readily available on the market. And I'm currently designing a new one with improved chemical characteristics. This is my design of very simple design, but results that we produce during research are really fascinating. I will mention some of them a little bit later. So this extractor slash reactor has practically pilot capacity, pilot scale capacity. This extraction reaction vessel has capacity 1.7 liter, and of course it must be made of highly resistant materials, because I mentioned that heated and pressurized water becomes extremely reactive. I want to explain that pressurization of extraction vessel, which is done through built-in valve on the lid of this extraction vessel, can be done with different gases for different, of course, goals. So this technology, cutting edge technology, is like a toy. You can play with it, achieve different goals in research and innovation. So depending on your goal and the treatment of the sample, if you target selective extraction of some phytochemicals with bioactivity or some other plant constitutes, you will pressurize, of course, with inert gas such as helium or nitrogen. But sometimes the goal can be decomposition of matrix, which sometimes has very complex structure. So reactivity is desired and can be potentiated by pressurizing this extraction vessel with carbon dioxide, which upon dissolution in pure water forms carbonic acid, which dissociates, of course, to hydrogen ions, giving a rise to acidity and actually acting as acid catalyst. The vessel is heated, and when extraction process is conducted, of course, it is very important to assure convective mass transfer in order to reduce diffusion layer at the border of our sample and solvent. So the extraction would become more efficient. So in this design, my design, I assured convective mass transfer by vibration movement of the platform housing this extraction vessel. So once again, when using water, subcritical water, so it's not water anymore because it has altered polarity, and in addition, other physicochemical properties are affected, such as surface tension, density, viscosity. They all are reduced with heating, which obviously contributes to very intimate contact with sample matrix, penetrating well into pores and assuring much more efficient extraction and higher yields. So many things can be done with pure water. My interest is to extract, isolate, separate valuable compounds from bio-waste. So in my research, I really managed with superior efficiency in comparison to other modern extraction techniques, such as microwave-assisted extraction, ultrasound extraction, to achieve much better yields and very characteristic chemical profiles, because simultaneously with extraction, some reaction with matrix constituents also take place, and some of these reactions, such as Maillard reactions, contribute to increased antioxidant potential of thus obtained extracts. So phenolics are, well, the most frequent phytochemicals that were extracted from different biowaste, because virtually any plant synthesize phenolics, of course, different polyphenolics, which have different bioactivities, anti-rheumatic, neuroprotective, cardioprotective. And the waste that was investigated, some of examples are given on the slide. Waste which has no other use, such as roseberry leaves, aronia bio-waste, aronia fruit is high in very bioactive polyphenolics, and the production, at least in Europe, has increased dramatically, leaving a lot of stems. Stems which in our research showed to be very bioactive. I mean, the extracts obtained by subcritical water. Also, interesting experiments. Again, we're talking about no limits challenges when using these techniques. So researcher can really express its creativity with this technology. Even omega fatty acids were extracted with water, of course, by adding some modifier. I opted for butanol, and this is the first case, actually, of using this modifier to extract, again, totally unpolar omegas, which are very important pharmaceuticals and nutraceuticals. Extraction of seaweeds. I will explain later that by using this technology, cascading approach in valorization of certain bio-waste or certain natural matrix can be achieved. Specifically, seaweed, brown seaweed, was obtained from Portugal, and the extraction was conducted with pure water. Those extracts showed very high content of different minerals, soluble fibers, phenolics. They were interesting to be used anywhere. And also exhausted biomass I used to develop functional cookies, because these fibers are also important, non-soluble fibers. And in the end, I want to also explain that this cascading approach, any sample exhausted with water can be treated under more rigorous conditions, approaching critical region to finally use biofuel from virtually any waste. So we can extract with water functional fractions, bioactive, use them in pharmacy, food industry, as nutraceuticals, cosmetology, then we can use remaining biomass for different purposes because biomass wasn't treated with organics, rather with pure water. So demonstrations of concrete results to confirm what I'm talking about. For example, chokeberry stamps, aronia. I don't know in India, but in Europe, it really gained popularity in recent years, let's say last decade. So many producers started producing large quantities, exporting to everywhere. But of course, stamps were underutilized. So by performing subcritical water extraction at, let's say, moderate, it is very important to choose adequate temperature. For this purpose, 130 degrees was chosen, and this table demonstrates bioactivity of waste extracts. So these stems extracts showed very good anti-radical activity, enzyme inhibitory activity, in sense that the extracts showed really high anti-diabetic potential, opening possibilities to use these extracts in some nutraceuticals because they are water-based, safe. Also, certain activities such as neuroprotective activity and activities important in skin aging. So important for cosmetic industry, or tyrosinase inhibition, meaning the extracts showed that have a positive effect on melanin skin pigment synthesis by inhibiting it, because it's also very popular today, this whole class of whitening cosmetic products. And ginger, very innovative result I obtained with ginger. Well, ginger, of course, it's widely used in India for different purposes. I approached it in different way. I treated it with subcritical water. As I mentioned, when using this fluid, it is very important previous experience to balance well its reactivity and solvating properties. So pungent compounds from ginger were degraded. So some derivatives were produced, not giving a rise to pungency of this extract. However, extracts showed remarkable bioactivity without having specific ginger pungency, ginger flavor. So we compared bioactivity, certain bioactivities, with conventional solid liquid extraction, and SWE, well, it is abbreviation for subcritical water extraction. This first column, of course, isn't subcritical because it is below critical point. However, we wanted to compare how activity changes with water temperature, because I mentioned that the temperature is key parameter. So anti-radical activity against DPPH radicals was the highest for extracts obtained at 150 degrees, and it is expressed as IC50. So it was quite low when comparing to conventional extracts. Total antioxidant activity and activity against hydroxy radical were superior for this extraction temperature of 180 degrees. Of course, other bioactivities of ginger extracts were tested as well, and this extract demonstrated also superior in comparison to conventional extraction, superior antimicrobial activity, both antibacterial and antifungal activity. In addition, anti-cancer activity of ginger extract was demonstrated in vitro studies involving 3 tumor cell lines. Of course, activity was also higher with conventional. So in these extracts, in addition to all compounds that are naturally present in ginger, we cannot neglect or ignore that probably neoform compounds also contributed, and also higher efficiency because more, well, richer chemical profile was certainly observed in subcritical water extracts. This extract was used to develop functional coffee substitute, and the results were published successfully. It was really innovation, again, in line with the topic and in line with these health issues, bioactivity, biotechnology, because we managed to actually offer coffee substitute, caffeine-free, with anti-cancer, antimicrobial, anti-radical, and other effects. So the extract, or let's say coffee beverage, coffee substitute derived from ginger extract, was sensory evaluated according to sensory properties by a panel, and all characteristics were evaluated, really rated really highly, and not a single member of this panel actually recognized that this coffee substitute wasn't of coffee origin. So our ginger-based coffee substitute beverage with anti-cancer, antimicrobial activity was compared with traditional coffee, with instant coffee, espresso coffee, and American filter coffee. So this is patent, really interesting patent. Of course, some other studies are required, but I would say it's an innovation. So when using this water as extraction medium, we should balance carefully its reactivity. Sometimes we choose extraction temperature to allow controlled hydrolysis in order to increase efficiency to actually hydrolyze analytes if they are bound to matrix constituents with very strong bonds. But when we want actually to degrade certain matrix, reactivity occurs and proceeds through different reactions. It is well known that in supercritical and subcritical water, not only hydrolysis takes place, but numerous other reactions, dealkylation, hydrogenation, cyclization, oxidation. All these reactions actually contribute to formation of new compounds that in some cases, well, alter both chemical profile and bioactivity of water fractions. So In my research, I try to balance well these water properties. In one term, hot compressed water properties, and it can be done a lot with this technology. I will just give few examples. For example, kernel of many fruits. I mean, we export a lot and produce a lot. Apricots, plums. Serbia is country of plums. So kernel remains underutilized, and it is also well known that the concentration of cyanogenic glycosides is quite high in the kernel. But with mild conditions and treatment with water, cyanogenic glycosides can be hydrolyzed, and mandelic acid, which is important platform molecule, important chemical, can be produced, again increasing value of this otherwise toxic waste of food processing industry. What I wanted to demonstrate with this table represents chemical profile of apple bark extracts, which were treated at temperature with water, subcritical water, at the temperature of 150 degrees. So actually, it is chemical profiling which confirms my previous statement that these extracts are very specific in their composition, and that compounds belonging to different chemical classes, different polarity, can be simultaneously extracted, and in addition, hydrolytical reaction and other conversion reactions take place. So in these extracts of apple bark extract, organic acids were detected, carbonyl compounds, alcohols, obviously acids, alcohols, sugars originating from sample matrix hydrolysis. This is polysaccharide hydrolysis, obviously, and formation of these sugars. So it can be used for the production of any of these sugars after separation. Even fatty acids were detected in pure water, but subcritical water extracts. And of course, this high solvating potential was also demonstrated with the presence of different compounds. And if I want to present more clearly, third stream of using this cutting edge technology, which I really love, is development of functional products, again, because we're dealing with water and water is safe. It is compatible fully with food products, pharmaceuticals, cosmeticals, etc. And I will just mention few research that I've done based on these extracts. Based on bio-waste extracts, of course, I developed different functional products, meaning from different categories, meaning products with add value. For example, in a very fruitful collaboration with colleagues from Portugal, Portugal is a wine producing country, but again, they have a lot of wine shoots that are not used properly because they can be valorized and their value can be increased. So from wine shoots, very rich extracts in sense of phenolic compounds and highly bioactive extracts in respect to their anti-radical and antioxidant potential was obtained, and cosmetic products from different classes were developed based on these extracts, gels, facial products, and others. And also recently, I extracted cocoa hull. Cocoa hull is also rich in many phytochemicals. So after obtaining this extract from cocoa hull, which is waste in chocolate industry, and it's used for burning, as a burning fuel, bedding for animals, so really not using it in proper way. I also developed a functional facial cream. Cookies with exhausted brown seaweed, I mentioned, and the advantage when extracting high water content bio-waste is that these materials mustn't be dried. Drying process is energy consuming, time consuming, and since we are dealing with water, this actually water content is beneficial. For example, it's not uncommon to produce biofuel from algae, seaweed, but in other technologies, usually it is required this drying process. However, with hydrothermal liquefaction, implying this technology, drying process is not necessary. Also, functional cookies, not like this, they were chocolate chips cookies, I developed from the extract of chestnut shells, and it is being well processed for publication. Very interesting work on chestnut shells extracts obtained with subcritical water, with very broad array of different bioactivities, good skin absorption of phenolics in that extract. So I incorporated around 1.7 of dry extract in functional cookies, and bioavailability was assessed. Lignocellulose biomass. I would like to briefly address this biomass because it's really widespread worldwide. Wood processing industry, and of course it is composed of different fractions, easily utilized fraction, cellulose and hemicellulose, and lignin, which is very stable biopolymer, not easily used by any technology. So we will talk and mention a lignin. So cellulose and hemicellulose are, let's say, already conventionally being converted to sugars, then by fermentation to ethanol, further to fuel, so many different ways. By using biotechnology, enzymatic hydrolysis can be achieved with these easily hydrolyzable, let's say, fractions. Of course, with subcritical water, it is also very easy to achieve, but lignin is stable polymer composed of isoprenoid units, and it's not easily depolymerized, degraded. It is really not easily degraded. As such, unmodified, undepolymerized, some applications of lignin have been proposed, such as using it as asphalt binder for lignin oil production, requiring very rigorous conditions, as a binder in certain composite wood products, etc. But
I'm sorry to interrupt you.
Yes,
ma'am, we have our next speaker has also joined us. So please, would you conclude the topic in 2 or 3 minutes?
2 or 3 minutes, yes, of course. So, in my research, it was treated at 180 degrees, and while the polymerization at very moderate temperature was very efficient. So we can see that total phenolic monomers and other monomers were very efficiently released. And also, the comparison was done in different atmospheres, nitrogen and carbon dioxide. So the optimal temperature for nitrogen atmosphere was 150, which was unexpected. Antioxidant potential was really high of lignin treated with subcritical water. Okay, so this is valorization of oilseed cakes, which were treated with water, and we managed to hydrolyze proteins because oilseed cakes are have very favorable chemical composition. Again, this is the proof that hydrolysis takes place very efficiently, and also they're rich in other nutrients such as minerals, polyphenolics. And once again, I developed cosmetic products, customized and very functional and very, let's say, interesting. So I managed to incorporate this knowledge in cosmetic products. Thank you for your attention. If you have any questions, I will be pleased to answer.
Extraction is required step for utilization of most natural bioactive compounds in food, pharmaceutical, and cosmetic industries. A subcritical water extraction technique for extraction of bioactive compounds has been of great interest in recent years. Thank you, madam, for delivering such a deep and insightful talk on how subcritical water can be used as an innovative tool for extraction, advocating the concept of green technology, which is at present the need of hour. I'm sure all of our participants would have been benefited through your words of wisdom. Thanking you once again, ma'am.
Pleasure.
Ma'am, any questions? I think, ma'am, the talk was self-sufficient. All our participants are contented with it.
Okay, if there are no questions, I wish you, well, successful continuation.
Thank you, ma'am. COVID-19 pandemic is surely one of the hardest experience we have all ever faced. The pandemic, termed as biomedical tsunami, impacted all contours of our life. It has given all of us important life lessons and at the same time highlighted the importance of technology and healthcare infrastructure. Post-COVID, there has been tremendous changes, particularly in field of medical sciences. To give us some more insight on the post-COVID era in field of biotechnology, we have with us our next eminent speaker, Dr. Sumit Sachdev. I heartily welcome you, sir. Before we move on, I present bio brief of Dr. Sumit. Dr. Sumit Sachdev is a healthcare compliance consultant with over 30 years of experience in Canadian and international healthcare. He is currently working as Senior Research Associate in the Office of Interactions with Industry at Mass General Brigham, Boston, USA. His area of work encompasses managing compliance with US Public Health Service regular Brigham policies to ensure that the relationship with industry do not compromise with patient care, research, or medical education. As a former physician from India, Dr. Sumit has been actively utilizing his unique background and experience to help physicians and scientists to navigate day-to-day legal challenges of medical practice and research. I now invite Sir to enlighten us on the topic, Exciting Future of Biotech in Post-COVID Era. The lecture that we are going to hear is a recorded lecture.
Hello. We are living in some very exciting times. With the internet and modern telecoms, time and distance are no longer obstructions to global collaborations. And thanks to global trade, practically everything is now available everywhere. Modern engineering has already taken us to the moon and beyond. And theoretical physics is pondering over the very birth of the universe itself. So why should biotech be anywhere behind? My name is Sumit Sachdev. I practiced as a physician in India till 2010 and managed a group medical practice in Canada till 2020. And now I have a front row seat in the real world theater of cutting edge medical research. And over the next 20 minutes or so, it will be my pleasure to take you on a journey to the future and introduce you to some of the fascinating advancements that are happening in the world of biotech research. It is my hope that even this very brief encounter will encourage you to make your own wonderful discoveries and give me something new to talk about at our next conference. Let's start with how long we live. These days, a newborn can expect to live for at least 60 or 70 years. Just a few hundred years ago, the average expectation of life at the time of birth would have been less than 30 years. Life expectancy at birth has more than doubled over the last 2 centuries. We are not only living longer, we are also living healthier and more productive lives. We owe this in large part to the advances in medical science and our increasing understanding of the natural forces around us. Of course, the advancing years bring their own problems. We are seeing more and more problems caused by accumulation of age-related DNA damage, most commonly resulting in cancers and neurodegeneration. The Human Genome Project is without a doubt one of the most astonishing achievements of our times. From the first description of the double helix structure of the DNA by Watson and Crick, some in the early 50s, to the ENCODE project 50 years later, our search for greater understanding goes on. ENCODE is an acronym for the Encyclopedia of DNA Elements. Genome researchers have produced vast amounts of data on the sequence of our DNA. And the ENCODE Encyclopedia organizes these data and makes it easily accessible to spur further research and discovery. And one of these discoveries is simply mind-blowing. Over the last decade, we have identified more than 23,000 genes. We have no clue what most of them do, but we have identified them. However, this accounts for only about 2% of the human genome. The remaining 98% is still a mystery. This mystery 98% of our DNA does not code for any protein. And at first it appeared that this was just a random jumble of nucleotides and was dismissed as junk DNA. But then researchers started seeing patterns. It turns out that 60% of a genome consists of repetitive sequences of nucleic acids. These are known as repeats. These repeats consist of many different families, ranging from small, simple sequence repeats to large arrays of tandem repeats to very complex mobile repeats that can transpose themselves to different locations in the genome. The origin of this mystery 98% can be traced back over the 150 million years of our evolution. Some studies even estimate that half of this, the dark genome, may be leftover sequences from long extinct viruses. Thanks to COVID, I think now even illiterate farmers in remote villages know what a virus is. But what is not as well known is that there are several such viruses called human endoretroviruses lurking in our own DNA. In fact, we already know that 8% of our DNA is made up of these ancient extinct viruses. Over the hundreds of millions of years of our evolution, these viral sequences have become an integral part of what makes us human. So the next time you come across a new virus, think of it as a long-lost cousin. In the biotech world, mysteries abound. Don't even get me started on the mitochondria, this indispensable resident of most living cells, the powerhouse of the cell, is still a mystery. I think except for red blood cells and some weird microorganisms, mitochondria are everywhere. And if you are a true mystery buff, you should also look into how octopi have been around for over 300 million years, adapted to radical changes without really evolving or significantly altering their DNA. To the curious mind, there is no dearth of wonder and amazement. From the lead character, her DNA, let us now shift focus to the supporting actor, the RNA, to the messenger RNA in particular. The success of mRNA vaccines for COVID has sparked a maelstrom of interest in what was often thought of as a sideshow. The rapid advances in biotechnology and molecular medicine gave us CRISPR. The CRISPR gene editing process has enabled us to harness this ubiquitous resource, and we can now imagine the production of almost any functional protein or peptide in the human body simply by introducing the appropriate messenger RNA as a vaccine or therapeutic agent. This has the tremendous potential to treat many, so far incurable diseases, and has ignited a frenzy of research in the pharmaceutical community. Diseases such as cystic fibrosis, metabolic disorders, several varieties of storage disorders, neurodegenerative diseases, dementias, and even several types of untreatable cancers are now targets on the firing line. So instead of using RNA, how about using entire cells for treating disease? Bone marrow transplantation has been around for quite a while now. What we were really doing was transplanting mother cells that would give rise to generations of daughter cells that would treat the deficiency. In other words, stem cells. Unlike blood, muscle, or nerve cells, which do not normally replicate, stem cells can replicate many times. When a stem cell divides, the resulting daughter cell could be either two stem cells, or a stem cell and a more differentiated cell, or just a host of more differentiated cells. We are getting closer to harnessing the signals that dictate this process. Now we can even take a normal cell and induce it to become a stem cell, the aptly named induced stem cells. Anyone fond of Marvel superhero movies? Well, my superhero has always been Superman. And my favorite Superman movies were the ones that starred the late Christopher Reeve in that role. In 1995, Christopher Reeve had an accidental fall from a horse, and that left him paralyzed from the neck down. He couldn't even breathe without a mechanical ventilator. Yet, he went on to become a real-life Superman and strenuously advocated for stem cell research. Thanks to his efforts and others inspired by him, we have come a long way from simply harvesting and transplanting marrow cells. Using the right cellular signals, once we have induced a cell to change into a suitable stem cell, the possibilities are endless and only limited by our own imagination. Induced pluripotent cells derived from tissue donated by sick patients are already being used to create organoids or organ-on-a-chip models for understanding the disease process. We can even test trial drugs on these models to find the perfect fit that will treat those diseases with minimal side effects. From these models of organs, the next logical step is to tell that cell to grow into an entire organ or even a limb. Current experiments have already yielded encouraging results. Researchers have learned how to grow heart valves, ears, and even the articular surface of bones by using chimeric scaffolds. Imagine implanting a heart valve in a newborn and then knowing that the valve will grow as the child grows. Science fiction may become science fact sooner than we can imagine. On a side note, research is also trying to grow human-compatible organs in humanized pigs and other animals. Just imagine, we may one day be able to order a Uber delivery for a custom-grown kidney or pancreas or even a liver. No more need for dialysis or painful insulin injections or having to stop drinking alcohol because of liver problems. But that's something to be hoped for. Growing up during the very early days of Doordarshan, one of my favorite memories is of watching Star Trek every Sunday morning. I don't know how many of you remember Dr. Bones from that series or his amazing tricorder. But we are getting close to that amazing device really fast. Artificial intelligence and robotics are making huge inroads into the practice of medicine. There are AI algorithms that guide diagnoses, screen for physician error, and may even create and direct treatment protocols. Many of the advances I have recounted have been made possible thanks to the use of AI or robotic applications. And as this technology grows, so will our accomplishments in medicine. Most of us have had the misfortune of having to get a blood test done and suffering through the lab techs' efforts as they probe around trying to find a vein. Well, we now have a robot phlebotomist that uses ultrasound to locate the vein and then with a single precise attempt slides the needle home. Technology that works. Efficiency at its best. And I'm sure you're already aware of the robotic surgical setups where the surgeon operates the knife from a remote location. This will be very useful on an upcoming trip to Mars. How about printing? When I was in school, simply getting a photocopy was an expensive excursion. Now printers are an essential tool and are found everywhere. But 3D printing is where we are headed. Industry is already routinely using three-dimensional printers to create everything from precision aircraft parts using metal alloys to printing entire houses out of cement concrete that you can just move into and start living in. So 3D bioprinters are not really that much of a stretch. The industry has now developed several forms of bioinks consisting of cells or bone matrix or collagen, and using these cells and biological materials as ink, these printers can print the structure or organ or even a functioning joint. The hope is that this tech will advance enough so that patients needing an organ transplant will no longer have to wait for a donor. Even modern telecommunications have a role. Imaging-based diagnoses was one of the early specialties to take advantage of the internet. X-rays and scans taken by a technician in North America are emailed to radiologists in India for analysis and reporting. How amazing is that? And speaking of imaging, I'm sure that at least some of you have seen an MRI machine. For those who haven't, the MRI scanner is a giant beast. It needs to be specially housed in several large rooms. It requires a dedicated electrical supply, air conditioning, the works. Well, a few months back, I was shocked to see a technician roll in a small portable MRI for an emergency brain scan. The device was smaller than a refrigerator, needed no special housing, and plugged right into the common wall outlet. Welcome to the future. There is a story currently circulating on the net that Sir Isaac Newton was working from home during the Great Plague when he discovered gravity and loads of other interesting stuff. Since we have also been working from home lately, and I hope that we will also discover interesting stuff, let me leave you with a quote from the sky. What we know is a drop. What we don't know is an ocean. So never stop exploring, my friends. Good luck and God bless.
Biotechnology, termed as science of genetic engineering, has assumed a greater importance with the advent of COVID-19 pandemic. The tiny coronavirus has brought terms such as stem cell, genome sequencing, messenger vaccine to limelight, and thus opening a plethora of research and development in field of medical sciences. The lecture by Dr. Sumit aptly reflected these ideas and at the same time provided an insight as how technology is advancing in this sector. A sincere thanks to Dr. Sumit sir from our side for the thoughtful lecture. We have with us Dr. Prashant Sharma as our next invited speaker for this technical session. On behalf of Sri Sathya Sai College for Women, I welcome you, sir. Without taking much of the time, I present a brief introduction of our speaker. Dr. Sharma is working as research scientist at University of Arizona Biomedical Campus in Phoenix, USA. Sir has completed his doctoral degree from Seoul National University, South Korea. Before obtaining his PhD, he also worked in research and development departments of a few biotech companies as scientist. He is a result-oriented professional with more than 12 years of experience in cancer immunotherapy, viral vaccine development, nanobiotechnology, artificial immunoorganoids, bioengineering, and project management across biotech industries and academia. He has also worked as research associate at the University of Wisconsin, Madison on cancer immunotherapy and humanized mouse model. He has also participated in a few WHO and Bill and Melinda Gates Foundation supported clinical trials while working at International Vaccine Institute for different vaccine projects for developing countries. Dr. Sharma has gained potentials in writing, revising, and reviewing SOPs, analytical protocols, analysis reports. He has reviewed more than 100 articles for various highly reputed journals. I cordially invite Dr. Prashant Sarma for his talk on the topic, zinc oxide nanowires are a potential candidate for intracellular delivery of biomicromolecules and cancer immunotherapy.
Thank you very much. Am I audible to all?
Yes,
Am
sir.
I audible?
Yes, sir.
Okay.
Yes, sir, you're audible.
Thank you very much for inviting me for such a great audience. And I'm because I belong to Bhopal, so it makes me a little better connect. So thank you very much. And as the conference is about the post-COVID era science and technology related things, so although I'll talk about my research as well, but talking about the post-COVID era, there are so many things which has changed. Change, like, see, we can still talk. I'm not there. I'm sitting here in Phoenix and I'm talking to you. This is a change came from Corona, because there was a lockdown for almost 3 to 6 months. And my lab also got affected. So we had to close down lab for almost 3 to 6 months and we didn't work. So we were working on some articles and things. And there are many other things the Corona has changed, like demand and supply. Like we are setting up a new lab and we ordered a liquid nitrogen cylinder. It took almost 9 months, like a period of a baby delivery to deliver. So there are so many things has changed. New technologies are also arriving. So I'm working on some special technologies, like we are developing artificial thymus. We are developing a humanized mouse model, but those projects are actually, we are trying to patent them. So we can't disclose those details here. So the research what I'm talking here is my previous research. So coming back to my presentation. I hope the slides are visible to everyone.
Yes, sir.
Okay, thank you very much. So, the topic of my presentation is Jadeno nanowire for intracellular delivery, but the point is utilizing a nanotechnology for the betterment of healthcare, betterment of vaccines, betterment of immunotherapy. Now, there was a little problem with the nanoparticles until the Corona came. So you people would be surprised to know that the vaccine, which is very famous for Corona, that's from Pfizer and Moderna, they both are using nanoparticles. So those nanoparticles, that mRNA is coated inside those lipid nanoparticles, and those are getting injected, and they have really high efficacy compared to other traditional first generation vaccines. So although it is the advanced, but we call it first generation mRNA vaccine because there has been no mRNA vaccines earlier. So it means if we talk about nanotechnology and apply that for the betterment of healthcare or the vaccine or related immunotherapeutic aspects, it would definitely work. So my topic, my research has been roaming around near those areas. So I have also included little part which we have recently published a paper. So what we did, we have stem cells, immune cells induced by nanotechnology for the cancer immune cells. So I have a very small part because my research is little longer. So my presentation would be in 3 sections. The sections 1, I'll talk about the intracellular delivery of biomacromolecule using the zinc oxide nanowire. The second part would be about cancer immunotherapy utilizing a little advanced version of nanowires, which I use for my section 1. And section 3 would be a smaller one, which we recently published utilizing the mesenchymal cells from fat tissue to make a artificial lymph node, and then ecotopically implanting it to mouse, and then checking if it can affect the immune system or whether it can, have the anti-tumor immunity. So as my most of my research is moving around zinc oxide. So zinc oxide is a metal oxide which most of the people might be knowing, and it is very commonly used for various aspects like photocatalytic applications, electronic applications, even the biomedical applications. So you people must be aware that these the sunscreen creams, they do have a zinc oxide nanopowder which help to protect from UV, and the cigarette butts has the zinc oxide which kind of protect the lung, but it's not possibly like completely protecting it from the side effects of nicotine. So if we talk a little bit about the past of utilizing zinc oxide in medical, so the first use we can see it was done by Charaka, and Charaka Sanhita is something a very ancient. I think everybody of you would be aware about it, and they use zinc oxide for some open bound treatment and eyes, and they named it Postpanjan. Then there's a lotion introduced by Greek physician Dioscorides. Then Avicenna, it's a Persian philosopher. He also used it for multiple skin diseases. So there's a big, if we go back in the history, then we can see that zinc oxide was used for different purposes, but we were not sure whether these were the nanoparticles or these were the normal zinc oxide powder. It could be, but the nanotechnology came existence a decade before. So now coming to the nanoparticle things, specifically biomedical applications, so zinc oxide has been applied for different things, like it works as a drug carrier, bioimaging, and many other. And it does have a very good antibacterial property, which we have also published research, which I'm not going to talk here. So coming back to the topic, this is the first. Okay, so you people see my cursor? You just can raise, somebody can raise a hand if I, okay, thank you. So this is the first report published from my lab back in South Korea. So what they did, they used a zinc oxide nano particle, which has a core of iron oxide. And they wanted to see whether this particle coated with the antigen can induce the immunotherapeutic aspects. So the best way for any immune system functioning, APC, it's called antigen presenting cells, need to be accumulated by the antigen. And once that antigen is inside the cells, then it can go and present the antigen to the T helper cells or CD8 or cytotoxic T cells, which can then go and kill the cancer. The same works for the vaccine as well, but for vaccine mostly works the prophylactic way. I'll talk about that as well. So they saw that if this can be taken up by the dendritic cells, so this, you see the green and red colors, these are the lysosomal and markers, plus the GBP is a peptide which is coated on these. So GBP is a very specific peptide which is coated on the zinc oxide nanowire. nanoparticle. So they saw it is taken up by the dendritic cell, and then they introduced these dendritic cells. It's called dendritic cell-based therapy. And they saw that there's a significant reduction in tumor. There's another report. What they use hollow Gedano nanosphere, and they use the, so in previous study they use the CA peptide. Here they use the OBA peptide. So OBA actually is a commonly used peptide which we can use it to express on the cancer cell. So these when they use these OVA, again they can be seen inside the dendritic cells in the lysosomes, and when they induce it, so here they work in both directions. Prophylactic means first you give the vaccine and then you induce the disease, and the therapeutic means first you induce the disease and then you apply the medicine. So that works in a both way. Okay, and so this little bit of history how people are using the zinc oxide for different kind of approaches. There's a good study from Dr. Deepak Shukla group. He's from Germany, and they have designed a tetrapod which they named Joton, and it is antiviral. So it worked very well if applied to the surface of vaginal area for the herpes, genital herpes, because what it does the same thing, because it has to work with for the immune system. So the APC should take up the antigen and then it could present to the T cells, and so you can see that there's a good results. Now my research is more about, like, I wanted to see whether they can go, but how they are gonna go and how it is gonna work. So there's a research, they have quantified, these people has quantified how many particles, how many nanoparticles per cell can be penetrated. So that was a good study. And here this is also my paper we published. So what we did, there's a very it's called neglected tropical disease. It's a bacterial disease which is called Orencia sutugomici, and there's no vaccine available for this. So we wanted to see whether we can utilize that nanoparticle to create a vaccine for that. So what we did, we isolated an outer membrane protein of a bacteria and then coated the protein on the zinc oxide nanoparticle and then used it as a vaccine. So when we injected it again, so you can see the, okay. So you can see this, there's a dendritic cell, and these, the red and green, red is lysozyme and green is the GBP peptide, which is having a tag of FITC. FITC is a fluorescence probe, which we can tag it on the peptide. And when we see this Z stack, it's a confocal imaging. So we can see it's merged. Merged means that the zinc peptide is going into the lysosome. And these lysosomes will then rupture it and present it on outer surface of the MHCs, and then they can work as a present the antigen to the T cell. So when it is presented to the T helper cells, which is CD4 cells, then it goes for prophylactic and it generates the antibodies for B cell. Like T helper cell helps for the B cell. And then, so we can see that there's a very good IgG1 and IgG2C response. IgG2C is actually the cytotoxic T cell response, and IgG1 is actually a Th1 response. And then we can also see there's a very significant interferon gamma positive T cells that is actually cytotoxic T cells. And we also check the we also challenge some mice after the vaccination, and we found that there's a good survival. So the survival rate was good. So it means that nanoparticle is working fine. And then there's a big question how these nanoparticles actually going inside the cell. It could be penetration or it could be endocytosis. So I have also worked on that. So this is just a slide to show that there are people, they use the one-dimensional nanoparticle and they wanted to show that most of the things are going with the endocytosis, not because of penetration. It's a little different than what I have observed, because mine was a 3D structure. Okay, so the second and most important thing in these kind of research is that some of these nanoparticles could be toxic, and these toxicity could, hinder the survival of specifically immune cells. And how it works actually, what it does, once the nanoparticle is taken up by the cells, it releases some zinc ion, and then it imbalances the equilibrium. And these zinc oxide could induce the ROS. ROS is reactive oxygen superoxide. So these can kill the cells, they can cause the apoptosis. But these all type of studies, they have done a single point study, which is not very beneficial to know exactly what is happening and what kind of cell, is it a apoptosis, is it a necrosis? So we need to work on that as well. So I started my study. This is my first, so that was some background, and coming to my first section of the my talk. So this is what we have developed. These are the nanowires, which with the help of Professor Young-Kyun Kim's lab from Korea University. So we divided these two nanowires and we named it two types. The first is called vertical nanowire. The second one is called fan-shaped nanowire. And these planes, so this is the first image you see, A, B, C, D, is a electron microscopy image. The second is, first is SEM image, the second is TEM image. And these are the peaks, which you can see is a very specific on this plane. So these the zinc oxide has a wood structure. So these peaks are very specific to zinc oxide. So we just confirm the purity. So we use these structures. And then we wanted to see how these, if we culture the cells on it, how they are going to react. So as I said earlier, the most of the toxicity assays, they have done single point study, but that study is not giving us any idea whether what is happening, how the cells are surviving. So what I did, I just cultured the cells and counted it for the different time interval and found that there's a little less growth, but cells are surviving. So I wanted to know exactly what is happening there. So then I did this apoptosis assay, and what we found that around 3 to 6 hours, there's a high rate of late apoptotic cells. Then slowly and gradually they start surviving. So it means if we see the necrosis, it's same as the normal cover glass, normal cells. So it means that the 60-hour, the zinc oxide has nothing to do with necrosis. It is mainly because of apoptosis, which is the transient one, which usually goes at the 3 to 6 hours of time interval. So this was a good study. Now we know that the cells are surviving. Now we wanted to see whether these cells how these cells are interacting with the cells. So, sorry. So this is actually the peptide which I use. It's called zinc oxide binding peptide, which I'll from now on I'll call GBP. So this is the same peptide which is published in Nature Nanotechnology from my lab. So I use the same peptide and coated it with FIT-C, and FIT-C is a fluorescence probe which we can connect with the peptide, and then I studied the binding. So if we see the fluorescence intensity and confocal imaging, you can see that within 64 to 128 micromole of quantity, it got saturated. So I was very happy because I just wanted to see how these the cells coated with these particles are interacting. So what I did, first I checked the scanning electron microscopy to see whether the cells are surviving and how the morphology looks. So if you see figure A, most of the cells are looking same. There's not much difference. Then I wanted to see exactly what is happening. So I stained those cells with a TOPRO, which is nucleus, blue color, and the cytoskeleton phalloidin, which is the red color. To my surprise, when I see that these cells are creating the philopodia, they are trying to catch the nanoparticles, specifically the fan-shaped nanoparticles. And there's a little bit of problem with the nucleus, because these nanoparticles are kind of penetrating into the nucleus. So that's something interesting. So we did jet stacking. Jet stacking means we can check the different planes of the cell, exactly how they are doing. So it's a jet stack. We have a different plane, and then we can merge them. So the figure in the bottom D is a G stack slide, and we can see that most of the particles is inside. The nanowire coated with ZBPFITC is inside the cell. So when looking at this result, I wanted to see exactly if these cells can take up the peptide from the coated on the nanowire or not. So what I did, I collected those cells once, first I coated the nanowires with the GBP FITC, which is a fluorescent probe, and then I cultured the cells for some 1 to 4 hours, collected those cells and did the flow cytometry. And we saw that most of the cells has taken up the peptide, which is a good thing. Now I wanted to know, as I told you, that there are two theories. Either the cells are taking the peptides or proteins through the endocytosis, or it is a penetration. So what we did, we did an experiment at 4 degree and 37 degrees centigrade. So 4 degree, usually the endocytosis got retarded. So there's almost no endocytosis at 4 degrees. So I cultured them and collected after a few hours. And when we see most of the cells has endocytosed that peptide. But there's a good number, like almost 60% in fan-shaped nanowires has also penetrated. So that's a really interesting result to show that the morphology of the nanowire or nanomaterial can change the uptake into the cell. So once we had that, we wanted to see whether we can also use the DNA and express it in the cell using this nanowire. So what we did, we use a PEGFPN vector, and then in that vector we cut the part which is called expression cassette, which does have a poly A tail and CMB promoter factor. And then in that part of DNA, I tagged biotin. So that biotin DNA, and then on the other side, what I did, I used the same GBP peptide, which is very specific to the nanowire. I cloned it with the streptavidin. Now I wanted to see whether these two biotin-related DNA, you might be very aware that this streptavidin biotin conjugation is one of the best conjugation available nowadays. So when we combine that, whether this DNA is combining with this protein or not. So I did IMSA. IMSA is the electromobility shift assay. So first I confirm whether these two, the DNA and the protein are binding together. So if you see, most of the DNA got bound with the protein. So that's a good thing. Now I wanted to see whether the protein is bounding on the nanoparticle or not, because the size is increased. So when I see almost like within 2 hours, most of the protein got bound on the surface. So that was a good results for me. Now what I did, I coated those nanowires, and here I use mostly fan-shaped nanowires because they have more transient penetration. So now I wanted to confirm whether these DNA can be expressed inside the cell. So what I did, I coated this nanowire with the zinc binding peptide, streptavidin, and biotin-related DNA conjugate, and culture the cell on it. And most of the cells, they express this DNA. So for the expression of DNA, you should the cell should avoid the DNA should avoid the lysosome, because lysosome, if it is in lysosome, it would be degraded. So it shows to the result what I have shown in earlier slide, that because of penetration it is happening, and there's no expression in vertical nanowire, which is evident that endocytosis, if it is going through the endocytosis, is gonna degrade the DNA, but because it is penetration, it works well. So this is the summary of my first slide. So there's a good endocytosis as well as penetration. It does form laminocordia, and then it expresses the DNA. Now, the same thing I wanted to carry forward for the immunotherapy. Now, the problem was that this nanowires were grown on the cover slip. So what we did, this is the second chapter of my presentation. So what we did, we discussed with my collaborators and we designed a special material, and these are PLLA membrane. So these PLLA membrane, polylactic acid membrane, and we coated the zinc oxide on that. So we call it PLLA ZNO. And the size of the nanoparticle was almost 5 nanometer. And then once they grow through the hydrothermal method, you can see there's a good structure is popping up, and this is the same figure, the peak, which confirms it's a good structure of zinc oxide. Now, the problem was we could easily either implant it for the therapy or prophylactic approach, or we can inject it. So the implanting could be little bit of cumbersome process, and it could be problematic for the mice as well, because we need to, you know, do the surgery, stitch them, and then give them the antibiotics and things. So we decided to go through the injection route. Now it was difficult. So what I developed a method to break down this membrane into a small fragments. So this is the process I developed. And then, so we do have a patent for that in Korea. And then we have 15 micrometers of size of these structures. Now, we did the same thing for the immune cells, the toxicity assay. To the surprise, the results are same. The only thing is that the apoptosis was little it's peaking at little bit of 3 to 5 hours of time period. Overall, the results are same. There's no necrosis, only apoptosis, but then slowly and gradually cells start surviving. Now, I confirm the binding. So here what I did, first I confirmed the binding with the GBP FITC conjugate, which we can see the intensity. If there's a zinc oxide, the intensity got very high, and only PLA, it's less. Now what I did, I isolated a protein which is very highly expressed on cancer cells, which is called CEA. CEA is called carcinoembryogenic antigen. Now, this carcinoembryogenic antigen is mostly expressed on the cells of cancer. So I cloned that CEA protein with this peptide and conjugated them. Now I wanted to see how this big structure is going to affect the binding. Now the PLA is again polylactic acid, has some non-specific binding, but once we add the zinc oxide nanoparticles on it, nanowires on it, it has the higher binding capacity. So if you see the binding of binding on PLLA is around like 14 to 15 microgram, but binding on zinc oxide PLLA is almost like 20 to 25. So that was a good result. Now I wanted to confirm how this structure is going to react in the APCs. So as I told you earlier, APCs are one of the most important cells for the immune system to work well. So what I did, I confirmed the uptake. So once the cell is, the APC is taking up the antigen, it will lyse this and then present on the MHCs, and then it will present to the T cell, which I have already explained. So most of the cells which has the zinc binding peptide has taken up the cell, and it is almost 90 to 95% of cell has taken up it. So it's a good result. Then we wanted to see how these dendritic cells are gonna react. So the dendritic cells, there are two types of dendritic cells. One is activated and one is non-activated. Once there is an activation of dendritic cell, there should be expression of some cytokines. There should be expression of some surface markers, which surface markers help them to go to the lymph node, present that antigen to the T cells, which help the T cells to go and attack the either cancer or virus, whatever. So when we see that there's a significant cytokine release compared to the other part. LPS is lipopolysaccharide, which we use for the positive control. So when we see even TNF alpha and interferon gamma are the very crucial cytokines which identified whether the immune system working is cytotoxic or not. So if we want to study the cytotoxic effect of any immune related drug, we need to study the TNF alpha and interferon gamma. And then we also confirm the surface expression of CD40 and CD86. These are the very crucial markers when the dendritic cell get activated. Otherwise, it goes down. So we compare with control and PLA, there's no significant difference. But when we check with PLA ZNO, LPS as a positive control should have very higher. Now, so then what I did, so I'll explain what I was talking earlier. So this is the antigen presenting cell. Once the either nanoparticle or nanowire is going inside, what it does, it activated the pathway MyD88 or IRAK through the TLR, and then if the zinc oxide is released, then like the zinc ions are released, then it will induce the ROS, which will enhance the cytokine, chemokine, and antigen presentation. Now this cell will go to the lymph node and it will present the antigen either to CD8 positive T cells or CD4 positive. CD4 is called T helper cells and CD8 is called cytotoxic. They are called cytotoxic when they release the interferon gamma. So what I did, I injected this complex like PLA ZNO plus ZBPCA to the mice, and after some 21 days, I isolated the spleen and checked CA specific T cell type. So if you can see, there's a significant increase in interferon gamma positive T cells, which is a very good results for us. Then what we did, we did the prophylactic as well as therapeutic effects, but we published the therapeutic aspect. So what we did, first we induced the tumor into the mice. So these tumors were having these, as I told you, CA surface antigen. And then once the tumor is little big enough visible, then we started the treatment. So we used the three week interval, and we injected this multiple times, 3 times. And to the surprise, most of the mice, the tumor got eradicated. There are very few, if you can see the figure, there are very few left with very small part of the tumor. So this, and then we also studied the specific lysis. So specific lysis, what we do is in vitro. So this is an in vivo result, and the if you see the B, it is in vitro result. So we culture the MC38 cells and then collected the spleen, induce them with CA, and then culture them for some time, and then remove the cells and then see how many cells it killed. So this is what it is. So now this was a good result. I wanted to know exactly how it is happening, what is happening inside the mouse body. So what I did, I collected the spleen of these tumor bearing mouse, and what we found that there's a significant reduction in T regulatory cells. Now the T regulatory cells are called immunosuppressive cells. So what they do, once there's a cancer, they will suppress the immune system. So if there's a reduction in T regulatory cells, it means it's a good result. So that's a very good result, that there's a suppression of T regulatory cells. There's a upregulation of CD4 and CD8 T cells in the tumor. And there's a good CD45 positive T cells. CD45 cells are considered lymphocytes. Okay. Then I also confirmed it into the tumor. So I collected the tumor, isolated the lymphocytes, and checked exactly what is happening. So it is very difficult to isolate and study the tumor infiltrating cells. So if you see, there's no change in CD45 positive T cells, there's no change in Treg, but there's a significant change in CD8 and CD4. It means these cells are going inside the tumor and they are killing. So this was my second part. So I summarize it in a picture. It would be easier for you people. Then comes to my third section of my presentation. So here what we did, we used the stromal vascular fraction from fat tissues, and then we made a organoid, and then we implanted into the kidney capsule with dendritic cell, which is a APC, which is already induced by antigen using zinc oxide. So this is, I kept it very short because it's a big paper. So I don't want to take too much of time, and I'll leave some time for questions as well. So here you can see I isolated these, like we isolated these cells. I'm one of the author in that paper, although I initiated this study. So this is the cells which we isolated, called stromal cells. Then we made the spheroid, and in this spheroid we use the same zinc oxide coated antigen, and this antigen would be taken up by the dendritic cell, very well known fact, and then we implant it into the kidney capsule, and then we induce the cancer. So you can see there's a significant reduction in cancer, and it functions as an artificial lymphoid, so artificial lymph node. So this is what we did, and this is the summary. So how does it work? There's a different kind of cytokine involved. There's different kind of factors involved. There's different kind of surface expressions of gene transformation is happening. So if you see this SVF, if there's upregulation of CCL21, which help for the T cell recruitment, if there's a upregulation CD40, CD80, it helps for the dendritic cell activation. And then once the T cell is there, then these T cells will go and kill the tumor. So this is overall summary, which I have already explained, that the cells can survive. There's a transient apoptosis. Biomolecule can go inside. These nanowire can efficiently deliver the associated antigen into the dendritic cells, and the SVF related things, which we know that it's one of the only study we have done right now. So this is all from my side, and this is the acknowledgement. So my PhD supervisors, these all my mentors. I work with Professor Namhyuk Cho in Seoul National University. Dr. Young Kyun Kim was my collaborator, and right now I'm working with Professor Mayo Otto, and he's a director in Phoenix Children's Hospital, as well as a professor in University of Arizona, and he's heading the cell therapy division. He's a director there. So this is all. Thank you very much, and I'm happy to take any question. Thank you very much.
COVID-19 pandemic has been a blessing in disguise as far as advancement in field of technology is concerned. The pandemic has ushered various new concepts in field of medical sciences. Dr. Sharma in his lecture beautifully highlighted how nanotechnology can be used in treatment of diseases such as cancer. I'm extremely thankful to Dr. Prashant sir for providing such a vast ocean of knowledge in a nutshell. Hope this lecture would be highly beneficial for our budding researchers. Thank you, sir. Celebrate endings, for they precede new beginning. With this thought, we come to an end of the first day of the 2 days international conference. Thank you to one and all for patiently listening and being with us throughout the entire day. Hope you all will join us tomorrow, as your presence matter and it will make a difference. Thank you once again. Sign up.